Yep. I kid you not.
At the bar, yeah. Hey, if you took
I think it's just people
honestly, that's so that's low-key what happened with mine, except
wing it along with me. Okay. So I don't know, is this on? Yeah?
Tomorrow is your dissertation day?
Okay. So I don't really have all that much to say. We are very happy to see everyone here today, and we have, I think most of the interns are in this little area over here. It's been 51 years since the first Harbor Branch summer intern came to Harbor Branch. It's quite a long time ago, so you guys are part of a long history, and I think we told you this on day one, a long history of students who've come here. And with this year, we have 17 interns, and it makes a total of 743 people who've come through this program. And many of them have gone on to very illustrious careers, and we know that all of you will as well. And we're really looking forward to your talks today. The primary sponsors for this program, it was really started with a gift from Ed and Marion Link back 51 years ago. And they continued to support this. And the primary sponsor is the Link Foundation. And the Link Foundation was founded by Ed Link. And I don't know what year. There is up here, anybody wants to know more about the Link Foundation, we have these little brochures, and especially for the engineering students, if you wanted to go on to a doctorate, you can apply for one of their very prestigious and great fellowships. So the students might want to pick up one of these. It's also on their website, of course. So the primary one is the Link Foundation, and then the other one is the Gertrude E. Skelly Charitable Foundation, and later on, Melanie Ciancoto, I think you say it, I probably said that wrong, will be here after lunch, or at lunch. And the other one that's been funding this recently is the Harbor Branch Oceanographic Institute Foundation, and they have funded one in honor of Marilyn Link. And we've told you before that Marilyn Link came to this up until her death. She was at virtually every symposium, and she would sit and ask hard questions, but sometimes she just asked, what are you doing next in your career? So maybe I'll be Marilyn today. Unfortunately, we don't have anyone from the Link Foundation who's here today. They weren't able to attend, so we had set aside some time for sponsor comments. And what we've decided to do is just move up the second session by 10 minutes. And so we'll get to lunch a little bit earlier. So if any of the people here have somebody who's live streaming it and or is going to watch the live stream, and you're in the second session this morning, feel free to text your families or whatever to let them know we'll be moving up about 10 minutes. We're very appreciative of the support of the sponsors, because without their support, this would not take place. And I want to say that this is a great year for us because we're supported by philanthropy. A lot of summer internship programs this year that are supported by the federal government did not move forward. They lost their funding. So we're very, very appreciative of this continued support. And we hope that the interns have had a great experience. Did you have a good experience? Yes. Did you have a bad experience? No. Don't tell us. You can tell me privately. We are really looking forward to hearing all about what you did and the projects. And I've seen some little bits of it as I was over here while people were practicing. So we're really looking forward to the symposium today. Esther, you want to add anything?
Just to remember that now you're part of this, you're part of the Harbor Branch family, you're part of your sponsors' family, and they take that very seriously. So stay in touch with each other, stay in touch with your sponsors, because they do appreciate knowing where you guys go. You know, we're all proud of you, and so we want to see what happens, and we're looking forward to your talks.
Okay, so now the first session is being chaired by Dr. Wayne Slade.
Good morning. I'm Dr. Wayne Slade. I'm here from Harbor Branch. Also adding to Esther's comment, keep in touch with your mentors too. We're very interested to hear how you proceed in life and your academic career. All right, our first session this morning is Engineering and Technology Application and Innovations. And our first talk is Gabriela Gomez from FAU. Her talk is going to be about aquaculture ecosystem modeling and aeration efficiency optimization using computational fluid dynamics. Her mentors are Dr. Bing and Dr. Wills, and her sponsor is the Link Foundation. So, come on up. That's the one from up here.
It's not on the... Oh. It's working.
Good?
Yeah.
Working. All right.
Good morning, everyone. My name is Gabriela Gomez, and I've been working on the aquaculture ecosystem modeling and aeration efficiency optimization using computational fluid dynamics. My advisors are Dr. Bing Yang and Paul Wills. Today, I will be going over the introduction, the hypothesis, methods, results, conclusions, and next steps of this project. Aquaculture is the practice of farming aquatic organisms, and it's very important because it contributes to the food network security, and it basically provides a good source of protein. Inland aquaculture accounts for 85% of fish production, and 90% of it comes from the production of from the pond systems. However, aquaculture has a challenge, which is maintaining healthy dissolved oxygen levels, which is very complicated and is critical because having good levels of dissolved oxygen will contribute to the fish health and the fish growth. So it is important to take into account the factors that affect DO levels, which are weather conditions, nutrition concentration, pond depth, circulation of the water, and also take into account the daily cycle of dissolved oxygen, which during the day, oxygens are high due to photosynthesis and the contact of the top surface with the wind. However, at night, they tend to lower because of the respiration from plants, algae, and bacteria. So in the bottom left corner, you can see a graph that basically describes the fluctuation of dissolved oxygen throughout the day. So a way to manage and have a healthy level of dissolved oxygen, it is important to monitor the levels using DO sensors and also focusing more on the levels throughout night, which is where it's critical. A way to mitigate the low dissolved oxygen levels, there are many solutions, but one of them is aeration. You could have fixed aeration or mobile aeration. Nowadays, like I said before, most of the lowest level of dissolved oxygen happens near dawn or early morning. And a way to challenge this or to tackle this, is to place the single mobile aerator where the pond is showing stress. But it would be good if there was a way to place multiple aerators that can tackle many zones of the pond. However, this is a very labor-intensive task. A way and a promising project is the AREPA project, which stands for Intelligent Resource Efficient Pond Aquaculture, which aims to control unmanned vehicles that will operate aeration and feeding. And the main question of this project is, will multiple low-power aerators be more efficient than a single high-power aerator? And basically, this is what my project was based on. I was trying to answer that question and see if configuration using multiple low-power paddle wheel aerators will distribute dissolved oxygen better than a single high-power aerator. And the way to do this, I divided the project basically in 3 steps. First, a geometry design, second, a 2D preliminary analysis, and finally, the 3D simulations. All the pond dimensions were gathered from a previous study. So I first developed a prototype with the recommendations. But for better computational analysis, I conducted a normalization using the depth of the pond and also a dimensionless analysis to calculate the velocity and the power, just for better modeling when using ANSYS. Something that is very important to take into account is that the physical pond has a leaf. However, for the initial model, it was neglected due to the size was negligible related to the overall pond size. So in this slide, you can see the 3D geometry. However, for the two-dimensional preliminary analysis, I extracted a slice on the x-axis, which you can see here. And the idea was to extract the slice where the boundary conditions, the aerator boundary conditions were going to be. And this was done so that I could focus on the vertical transport, also fine-tune the simulation setup before scaling up to 3D, and to select the appropriate physical models. In this video right here, you can see how the mobile aerator works. And this is basically what we were trying to mimic with the ANSYS simulations. This type of mixing and circulation, also oxygen injection in the pond. And to the right, you can see the 3D initial pond geometry that was used for the simulations. This slide basically shows the setup for both the two-dimensional and the three-dimensional simulations, where we compare a single aerator and a multiple aerator configuration. For the single aerator, you can see that the first parameter is the velocity. But this is basically representing the power capacity that we were trying to apply on the boundary. Our geometry, the prototype, was a total of 10 acres, and our goal was to have 1 horsepower per acre for a total of 100 horsepower. So the single aerator was going to have a capacity had a capacity of 100 horsepower. And with the dimensionless analysis, this correlated to 3.36 velocity in the x direction. And to test and basically see the efficiency of both of the configurations and compare them, for the multiple aerator, the idea was to use the same horsepower but divide it into the amount of aerators that were going to be placed in the pond. So in this case, because this is the setup for the two-dimensional configuration, is just 2 aerators, so each represent 50 horsepower for a total of 100 horsepower. With the dimensionless analysis, each represents 1.68 velocity in the x direction. It doesn't have units because, like I said before, it's a dimensionless analysis. It just represents velocity in the x direction. For the second parameter, we have the specific flux of oxygen. This represents the aerator boundary condition, and this is the equation for the oxygen input. And the last parameter was the shear stress, which basically represents the free surface of the fluid. For the 3D simulations, like I said, we first did a single aerator configuration and then a 6 aerator configuration. Right here, this is the aerator boundary condition that represents the 100 horsepower. And for the 6 aerator configuration, they're placed on the edge of the pond and each represent 1/6 of 100 horsepower. And the bottom images, basically, these are the mixing patterns we were trying to test and see which one, if either the single aerator or the 6 aerator was most efficient. For the results, for the 2D preliminary study, we aimed to basically have a boundary condition that was inputting oxygen into the surface. So these images or these contours you see right here were the goal, and we were able to reach it. And we did this in order to then scale it up to 2D. So the simulation and the setup for this 2D simulation was also used for the 3D. When I was able to verify the equations and verify that the flow was correct, I used the same simulation for the 3D. And I ran the simulation for 5 minutes, real time. And these are the results for the single aerator simulation. First, this is the velocity magnitude contour top view. I then extracted the results from ANSYS and wrote a MATLAB code that processed that data and outputted a sliced contour, basically just to focus on how the boundary condition was affecting the depth of the pond. So you can see right here, this is the singularator, and this is how the velocity magnitude of the flow looks like. Then these are results from the same simulation, singularator simulation, for the same 5 minutes, but this is the mass fraction distribution of oxygen. The slice contour where the yellow zone represents where oxygen is, and to the right you can see the mass fraction top view of those results, where if right here where would be where the aerated conditions are. Then these are the results for the multiple aerated setup, but this is the velocity magnitude. To the left, the sliced contour, and to the right, the top view of the velocity magnitude, where you can see the flow coming out of the aerated boundary conditions. And for the same simulation, the same 5 minutes for the multiple aerated simulation, to the left, you can see the sliced contour with localized oxygenation, and to the right, you can see the top view of that same simulation. The key insights from these results are basically that multiple aerated configurations tend to produce more localized oxygenation with better surface coverage, but a single aerated configuration tend to cover a large volume quicker but less precise. And another key insight is that the flow patterns from the paddle wheel boundary conditions appear inaccurate and need more debugging and correction. So this is why our next steps in this project is conducting a dye test to capture those flows and then calibrate it to the model to have accurate aerated systems. Also, we want to test different parameters and see how that can change affect the effectiveness of each configuration. For example, different power capacities for the multiple aerated systems. We did 1/6 in this case because it was 6 aerated conditions. We did 1/6 of the power, but we want to try different capabilities and also different distribution of power. We want from these results to compare the surface coverage and average coverage, the average concentration, just to see which one is more effective. Also, optimize longer simulation runs. And one that is very important is to compare the time to reach a specific mass fraction of oxygen for each of the configurations, which would give us more insight into the effectiveness of each configuration. Finally, I would like to talk about a last task I did while running the simulations. So this is the pond bathymetry using LiDAR data. The goal with this is to fly a drone with a LiDAR pointing downwards to catch the geometry of the pond, which would be used for the model to model the predicted pond ecosystem. And the first test we ran was basically using the Aqua North parking lot. We flew the drone on top of the objects you can see over here, the tank and the dumpster. And the idea was to catch the altitude data and process it with a MATLAB script to get this surface plots right here. You can see this is the dumpster and this is the tank. So these are the first steps before we move it to taking the drone to the fish farm, and gather the data for the geometry of the pond. Finally, I would like to thank the faculty, graduate students, and fellow interns, and also Harbor Branch and Link Foundation for this opportunity. And that'll be all. Is there any questions? Thank you.
It's very interesting to see the void, where the voids are, and where the concentration is, but I'm also wondering how
So this is, the idea is in the future to have a model that would include the species, because the idea is to have a predicted model using ANSYS. And from the studies I've done with ANSYS, you're able to include species into that. But right now, this is like a preliminary study. Right now we're focusing on seeing how the aeration is going to affect the flow of the pond, basically. Yeah, so let me see if I can go back. So from this video, this aerator is attached to a tractor. So all of the aeration, the idea is to have the multiple aeration systems attached to a tractor that is going to place the aeration systems where the DO levels are low. So from this slide right here, the mobile aerator configuration or the mobile aerator system is what we're using, but the ARIPA project is using unmanned tractors that would place the aeration systems where it's needed or where the pond is showing stress. Okay, I understand now. It should be possible, but we will have to be like a vehicle or some type of aquatic vehicle that would just place the aeration system where the DO levels are needed. But right now, the project is using the tractors. You're welcome.
Thank you very much. All right, our second talk this morning is Paige Keep, also from Florida Atlantic University. She's mentored by Dr. Adi Nayak and myself, sponsored by the Maryland Sea Link Memorial Internship, which is part of the HBOI Foundation. Her talk today is going to be development of a cost-effective holographic imaging system for plankton studies.
Okay, good morning. My name is Paige. I worked with Dr. Nayak, Dr. Slade, Ahmad Abdullah, and Karuna Agrawal this summer to fabricate the design of the cost-effective holographic imaging system, the HoloPi. A quick outline of what I'll cover today is the motivation for my project with the importance of plankton and traditional sampling methods, or sampling practices. Then we'll look at one method for plankton imaging, which is holography. That will include the AutoHolo and the HoloPi. Then we'll look at the development and construction of my project, which includes the optical and imaging components, as well as the internal design. And then we'll finish by looking at the future plans for my project. We study plankton because they greatly influence ocean and human health. Plankton are at the base of most aquatic food webs, making them vital to the well-being of countless marine organisms. Plankton also drive the carbon transport, which is the capturing and storage of excess carbon dioxide. This in turn helps regulate CO2 levels and the effects of climate change. The abundance of plankton have been tied to causing harmful algal blooms, or HABs. Lastly, the distribution of plankton fields have been seen to affect how light and sound propagate through the water, which affects remote sensing projects and the overall function of the ocean. So some of the traditional sampling practices involve the use of nets and bottles. The issue with nets and bottles sampling is that it's very manually intensive and time-consuming, as it requires lots of in-lab enumeration. A different example of plankton sampling involves the use of an IFCB. This uses flow-through technology, which often breaks up the particles, can miss fine-scale layers, and doesn't capture fluctuations. Overall, these sampling methods are not a good representation of how the particles act in their environment. That's why we are looking for a new and improved method that is free stream and in situ. This innovative method is digital holography. In basic terms, holography utilizes a laser to illuminate the sample so that its contents can be photographed. There are two types of holography: offline, off-axis, which is the top image, utilizes mirrors to split the laser beam. Off-axis generally gives us better resolution of holograms, but compared to the second version, which is inline, the inline is preferred because of its simplicity. Both require collimation of the laser beam. So collimation basically means that the rays are parallel to one another. So this image is showing uncollimated light versus collimated light. This next illustration is showing how holography illuminates the sample volume to create a hologram, and then this hologram is reconstructed to give us the 3D data. So overall, holography can give us accurate and advanced 3D information like the position, size, and shape of plankton. One application of digital holography is the AutoHolo. This is an autonomous in situ imaging system custom built here at Harbor Branch. The two images on the left are showing a tow-behind application and deployment of the AutoHolo. The next diagram is showing the two cylindrical housings, which hold the camera and laser, respectively. The cylindrical housings measure about 13 centimeters in diameter and about 61 centimeters in length. The biggest advantage of the AutoHolo is that it's an in situ imaging system, and it uses free stream technology so that it's non-invasive to the plankton. You can also adjust the sample length so that you can control the size of the sample volume. Additionally, the AutoHolo takes very high resolution images with a 4,920 by 3,280 pixel camera. This CopePod hologram is an example of an image taken by the AutoHolo. There are some challenges to the AutoHolo though. It's very expensive. Parts alone cost $30,000. It's also very intensive to build, meaning it's hard to scale and replicate. These drawbacks is what has prompted us to find a new frugal imaging system. So the goal of the HoloPi is to be affordable at $3,000, just 1/10 the cost of the AutoHolo. It will have accessible off-the-shelf parts, and it will have a small footprint. It will have about 7.5 centimeter diameter, and each housing will be about 30 centimeters in length. It will also be under 15 pounds. This image is a basic idea for the HoloPi. You can see the camera side and laser side are in line with one another. The sample or the depth sensor, the pressure valve, and sample volume are also pictured. So the overall hope is that a cost-effective holographic imaging system will advance and increase plankton research. So in order to create a cost-effective holographic imaging system, these are the optical components we will use. We used a 532 nanometer green laser, which first passes through the neutral density filter so that the strength of the light is reduced. The beam is then collimated through the objective lens, pinhole and translator, and the diverging lens. Once the beam is uniform, expanded, and noise reduced, it passes through the convex lens to be condensed and to illuminate the sample. So my focus this summer was to design a structure that houses all these components in an adaptable and durable way while still allowing for proper use of each component. This was the internal structure I came up with. I used a 3D modeling software, SolidWorks, to fabricate this design. The image and animation shows how each component aligns with one another and is adjustable, yet each piece is protected and the overall cage system is very sturdy. My biggest challenge this summer was the tedious constraints with each component and the limitations with diameter and size. I also had to make it flexible for future use. The next piece of the HoloPi is the imaging components. The fabrication of this side isn't complete yet, but this is our working design. We utilize a lens and camera global shutter camera with a 1,456 by 1,088 resolution. We also used a Raspberry Pi as the computer board. There is space to expand the computer board and space for extra electronics like sensors. As of right now, the cost for all the components is about $1,160. So that means we are on track for achieving our goal of creating a cost-effective holographic imaging system. The technique I used to construct this low-price structure was a 3D printer, specifically the Bamboo X1E. I used ABS GF filament, which is extremely strong and durable compared to other 3D printed plastics. I used threaded inserts, which I essentially melted into the plastic using a soldering iron. These are beneficial because it allows for repetitive use of the 3D printed parts. This was the final iteration of the laser side that I 3D printed. It's currently in the lab for testing, but this is the first iteration that I have. As you can see, the pieces move, and then you can tighten them with the screws and inserts. The other results of my project this summer focused on verifying the accuracy of the optical components. So this means verifying that a collimated beam is achievable. Again, collimation implies that the laser beam is uniform in strength and diameter at multiple distances. So this first image is about 12 centimeters away. You can see that the green laser is uniform in strength and has a diameter of about 2 centimeters. The second distance is about 90 centimeters away, and again, it's uniform in strength and the diameter is about 2 centimeters. We were also able to get a preliminary raw hologram, which is the far right image. Yay. The future developments of the HoloPi include reconstruction of the hologram, so taking that last image and making it cleaner, basically. Then construction of the camera side, so printing the parts and adding all of the electronics. The overall goal of the HoloPi is to increase the global use of holography for plankton studies. This could increase long-term monitoring of ecosystems and can minimize the negative results of HABs. I'd like to gratefully acknowledge my support from the Maryland Sea Link Memorial Fellowship and the Harbor Branch Oceanographic Institute Foundation. Thank you, Dr. Nyack and Dr. Slade and the rest of Nyack Lab for all the mentorship and support this summer. And a special shout out to Addison for being a great friend and role model.
Time for questions?
Thanks for a very interesting presentation. And, I suppose actually your last thing that you said is how this could help to mitigate the effects of HAB. Could you elaborate on that a little bit more?
Really interesting because the next presentation is kind of going to go into that a little bit more. But basically, like I said, plankton have been seen to, when they're in extra abundance, they're what causes HABs. And so if we're able to monitor the number of plankton in real time, we can kind of see if they're starting to increase and hopefully take action quicker, so that we can minimize HABs. But I wish I knew more of the marine biology side, but hopefully the next presentation will explain a little bit more about that. Thank you.
I have a question. How sensitive is it to alignment between the camera and the image system?
Very sensitive,
How
yes.
would you evaluate that?
The alignment does take a lot of time. I tried to help but learn some about the alignment, although my mentor, Ahmed Abdullah, was definitely the one that was aligning all the optical components, but they are very sensitive. So you have to account for the focal length with each piece, and then again with the translator and pinhole, making sure everything is aligned with one another. So it is a very tedious process.
Tedious, good word for it.
Great presentation.
Thank you.
I guess my question is, are you going to run both the camera side and the laser side off of the same Raspberry Pi board, or do you have to coordinate multiple boards within your housing?
It is through the same board. The Raspberry Pi will be running the global shutter of the camera, and then it will also be telling like the laser when to run, I guess. So it is all through the same computer board.
That's it? Oh, we got one more? Oh, yeah.
Yeah, I have a question. It's about the water sample. Sedimentation of high turbidity affecting the source in any way?
I'm sorry, say that again.
Sedimentation of high turbidity in the water sample affecting the source in any way?
I'm not sure. I believe that if there is sediment in the water sample, when you take the hologram, when you reconstruct the hologram, that's where you're able to filter out unusable holograms or holograms that are picturing plankton particles like plankton. So the reconstruction of the hologram is where you can filter out like information that you're not wanting, I guess, if that's answering your question. Okay.
All right. Thank you. All right, our third presentation today is Addison Ryan from California State University, Monterey Bay. She's going to be talking about advancing rapid quantification of microcystis colonies in freshwater systems using digital holography. Her mentors for this were, again, Dr. Adi Nayak and Dr. Malcolm McFarland, and her sponsor is the Link Foundation.
Awesome. Thank you so much. Good morning. My name is Addison. I spent the summer in the NIAC lab contributing to some work in an ongoing project that is using digital holography to estimate cell counts from images of individual algae colonies. So the species that we were studying is called Microcystis aeruginosa. It's called a blue-green algae classified as, but it's technically a colonial cyanobacterium. Its cells secrete this sticky mucus-like substance that causes the cells to stick to one another and clump together in groups called colonies, and those look like this. These colonies can then rapidly grow on the surface of bodies of water in what's called a bloom. These blooms are becoming more frequent and severe, especially in freshwater lakes. Some of these blooms produce toxic microcystins, which are hepatotoxins. They can consume all of the oxygen in the water, kill aquatic plants and animals, contaminate the drinking water, and threaten human well-being. These toxic blooms are called harmful algal blooms, or HABs. Because of their dangers, monitoring HABs is incredibly important, and that means counting cells from colonies. Cell counts are the actual units used to assess water quality and predict bloom potential. Mitigating a HAB is not just about measuring chlorophyll levels or imaging colonies, but getting actual cell counts that contribute to management decisions. This study uses 2 sample strains, one from Lake Erie in Michigan and one from Lake Okeechobee here in Florida. To count phytoplankton like Microcystis using traditional techniques, you'd first have to obtain the colony using something like this net, and then break the colony apart, sonicate it, possibly chemically stain it, and finally use a flow cytometer or microscope to get that eventual cell count. This process is very slow, it destroys the colony's structure, and it's not field friendly, meaning you can't do this on a standard boat. Additionally, anything that we perform in a lab will not accurately reflect the dynamic ocean conditions that are constantly changing. This is where the NIAC Lab's AutoHolo system, developed here at Harbor Branch, comes in. It's a submersible version of a digital holography setup that can be operated in situ to image aquatic organisms, like the 2 kinds of salps pictured on the right. These were just collected in a field deployment off the coast of California in June. It collects 3D images of intact colonies as they flow through the water, without needing to disaggregate or transport them. The challenge is then turning those images into ecologically meaningful data. So instead of focusing on just detecting microcystis colony presence, our goal is to ask, can we extract enough information from a hologram image to predict how many cells are inside a colony? To answer this question, we first isolate a single intact colony and image it using digital holography. This produces a 3D image stack similar to a CT scan by reconstructing how light diffracts through the colony. Then we sonicate that colony to break it into cells that are individual. that can be imaged holographically, as well as validated by traditional methods, which are represented in the darker boxes. These methods include being run through a flow cytometer, which uses fluorescence to count the number of microcystis cells in the sample, giving us our ground truth cell count. We can further validate this number through traditional microscopy by manually counting the number of cells in a sample volume. After an experiment has been run, we then use MATLAB for hologram reconstruction and extracting morphological trait information. We also use a technique called confocal microscopy to image full colonies outside of the experimental workflow. This approach uses lasers to create 3D images by scanning the sample point by point across various depths. This gives us important information about the colony's structure and is used to determine the average colony thickness for each strain, which we then incorporate into our MATLAB processing codes. Ultimately, we're trying to link what a colony looks like to how many cells it contains to be able to predict one from the other. The multiple tools together help to build the full picture. Digital holography gives us volume, colony shape, and porosity. Confocal microscopy provides the thickness and internal structure information. Flow cytometry gives us a cell count and the ratio of live to dead cells. And traditional microscopy lets us double-check all of our counts manually. Here's how the imaging actually works. Paige gave a great setup for it, but in the lab we use a 632 nanometer red laser, which gets filtered and collimated to produce a clean, uniform beam. That light passes through the colony sample on the slide and through a 5x objective lens. It interferes with itself as it passes through the sample, and that interference pattern is captured by the camera. The resolution of the resulting image is between 1.64 and 1.66 pixels per inch, and you can see an example here from each strain. We then reconstruct those patterns into a series of depth planes using MATLAB. It's like creating a 3D X-ray of the colony without having to physically slice or stain any of it. It also lets us see the structure then all the way through the colony in a completely non-invasive way. This slide shows exactly what that looks like. On the left, you see a traditional microscope image. In the middle is the holographic splat, which is a 2D projection of all in-focus layers. On the right, that same image has been processed with binary segmentation after isolating the colony from the background. From here, we can extract traits like volume, solidity, and hole ratio, which lets us go from a blurry blob in a laser to something measurable. The top row is the Lake Erie strain. These colonies are dense and compact like a slab. The Lake Okeechobee colonies on the bottom are more porous and irregular, like a sponge. This summer, I performed 39 experiments and processed 20. Combined with historical data, this gave me a total of 41 data sets to analyze and apply our predictive modeling. Let's start with the Lake Erie strain, since this is where the model performs best. When we plot colony volume against baseline flow cytometry cell counts, we get a clear linear relationship with an R-squared of 0.71, meaning that volume explains over 70% of the variation in cell count. This is a very strong predictive signal, especially for biological data with lots of inherent noise. It tells us that as volume increases, cell count increases as well in a predictable, statistically significant way. You can see how tightly the blue points fall along the line. This is what gave us the confidence to build a predictive equation from this relationship. Using the linear regression, this equation is the empirical relationship that we were aiming for, where the estimated cell count equals roughly 1.65 million times the colony volume minus around 39,000. That might seem like an unreasonably huge number, but this equation is scaled for 1 microliter, whereas most colony volumes are less than 0.3 microliters. If we instead plug in a representative moderate-sized Lake Erie colony volume of 0.06, the equation predicts around 60,000 cells, which matches very closely with the results that we got from flow cytometry and holography for a colony of this volume. So this isn't just an abstract model. This is a practical tool that gives us biologically realistic cell counts using only colony images. So why does this equation work so well just for the Lake Erie colonies? A big part of it is internal consistency, specifically solidity, which measures how tightly packed the colony is by dividing the actual volume of the colony by the volume of its outline. Lake Erie colonies had significantly higher solidity, around 0.96 on average, compared to 0.82 for Lake Okeechobee. Lake Erie colonies tend to stay dense as they grow, which is why volume continues to reflect cell abundance. The Lake Okeechobee colonies, on the other hand, become more porous as they get larger, contributing to the wide spread of solidity values that we see here. So volume only works as a predictor when what's inside the volume stays consistent. When we separated out the Lake Okeechobee data for larger colonies, we saw that the relationship between colony volume and cell count is positive for small and medium colonies, but negative for large ones. This tells us that volume may start out as a useful predictor, but as colonies get bigger and more porous, the relationship weakens and eventually reverses, so volume alone becomes incomplete as a predictor. Once the colony structure starts changing, we need more than just size to understand what's really happening inside. We saw this morphological difference observationally throughout the project. Lake Okeechobee colonies were denser at smaller sizes, but began to diffuse at larger volumes. This change in structure is exactly why the relationship changes, and volume is no longer reliable as the sole predictor of cell count. Again, it's not that volume doesn't matter, it's that the volume to cell count relationship changes in a way that volume alone can't capture. We need a model that adjusts for the ways that colonies actually grow and change. This plot helps to visualize the relationship between colony volume and solidity for both strains. For the Lake Erie colonies, it's simple. Solidity stays high and consistent even as volume increases. So for our model, a larger volume correlates with more cells in a tightly packed structure. But for Lake Okeechobee, solidity is much more scattered. Some colonies were over 90% solid, while others were only 3%. So two colonies from the same strain could have completely different internal structures and wildly different cell counts. Those internal structures contribute to prediction bias in holography. As colony volume increases, holography starts to underestimate cell counts for Lake Okeechobee compared to flow cytometry. This reinforces what we've seen with the prediction strength of volume and tells us that while holography isn't failing, we need to account for the colony's overall structure to be able to interpret it correctly. So even though we saw some prediction bias and challenges with the Lake Okeechobee strain, holography is still a very reliable method for obtaining cell counts. When we compare it to flow cytometry, the two techniques have strong agreement overall. The difference between cell counts of the two methods was around 35%, which isn't perfect, but for imaging intact colonies in 3D, it's a very strong start. The Bland-Altman plot helps us see this relationship. Most points cluster around the center line at 0, indicating that holography and flow cytometry counts are often very close. The spread only starts to widen at these higher cell counts, as we would expect, but with only a few points outside the expected range of variation indicated by the red lines. So even though holography does have some bias, it's still very closely aligned with our standard cell count method, and that bias is something we can account for, not a deal breaker. So what does this all mean? First, colony volume is a strong predictor of cell count, but only when the internal structure is consistent. In strains like Lake Erie, where colonies stay dense as they grow, we can make accurate predictions using just an image. Morphology, especially solidity, contributes to when volume-based prediction models fail or succeed. For Lake Okeechobee, volume isn't enough because the colonies diffuse as they grow, breaking the relationship between size and abundance. Digital holography is a promising tool for in situ cell estimation, but it needs morphological trait calibration to account for structural differences. Instead of applying one equation to all colonies, we'll need to adjust the model based on thresholds of size, solidity, or porosity. And that's the direction this work is heading. With morphological trait calibration, we can use holography for in situ quantification of microcystis cells with no disaggregation, no toxic dyes, and no manual counting. This would hugely advance fast, scalable HAB research. We've processed 41 colonies so far. I have 19 more holograms already imaged and ready to process, which will bring us to 60 total colonies to better test prediction thresholds and increase statistical power. We anticipate the additional unprocessed Lake Erie colonies will fall more within this mid to high range of volume and solidity, which is currently underrepresented in our data set, so that should help refine our equation. The AutoHolo has also already been deployed in Lake Erie for repeated field efforts over the past few years by NIAC Lab PhD student Ahmed, giving us a body of holograms that we can now apply our empirical relationship to. This brings us one step closer to getting real-time cell count estimates in the field from holographic colony images. I'd like to thank Dr. Nyack and Dr. McFarland for their mentorship, Madison and Olivia for fast-tracking my training before their crews and cheering on my success, Dr. Karuna Agarwal for designing the project and helping me learn a new coding language, and the whole optics lab team for their support. This project was funded by the Link Foundation, and I'm grateful for the opportunity to contribute to their work. I'm also forever grateful to the other summer interns, especially the other NIAC Lab intern Paige, who became not only a great collaborator, but a great friend. I really appreciate your attentiveness, and I'd be happy to take any questions.
We have time for questions? Zach.
Congratulations, that's a really interesting result and cool project. I guess I'm wondering, you have these two different, growth kind of modes of the same organism. Why, could you speculate as to why that might be? Why do they grow so much denser in like really?
It's a very interesting question. For anyone who couldn't hear, the question was about why the two strains develop so differently. There isn't really a great answer for why different strains of the same species would have so much morphological difference. What we do know is that Microcystis is a worldwide species. It's found in freshwater systems all over. So there are probably traits that we're not even accounting for that might be important. There's a lot that goes into like the population dynamics. They experience a die-off at the end of the summer, pretty much every summer. So there are just, also the fact that one of them's coming from Michigan and one's in Florida. So the actual like ecological distinction of their two habitats is also very diverse. So I just expect that the species are as diverse as the areas that they're found in. Mm-hmm.
I can't help with the answer. It's about nitrogen phosphorus.
Thank you.
That affects their, like the polysaccharides?
Yeah.
That kind of stole my question. I have another question. How sensitive are the results to the method that's used for the binarization of the imaging of the holograms, so the segmentation?
So the segmentation, we go through a whole coding process. The actual MATLAB processing, for me, new to MATLAB, it takes about 3 hours to get through a single colony from one experiment. And that's because we go through and we're able to compare, we change thresholds like decimal by decimal and compare until those images are pretty much exact. So it is subjective to the person running it, but we go through the process of making sure that those are as closely related as possible. Yeah.
Dr. Moore.
Yeah, very nice, very
Thank you.
interesting. Were these based on cultured colonies or field samples?
These were based on the cultures. So we have the tubes and then we work out of big bottles too.
And they have the same kind of growing conditions?
Correct, here, yes.
Okay, so the differences are sort of internal to the species or subspecies, I guess.
It does seem that they're strain specific, and I will also toss out there that this is not something that was part of the project, but we'll see differences within the same strain in the same body of water. So that's just why this approach, if we can get something nailed down that's specific for each strain or each morphological type, instead of just applying one equation, that will allow us to kind of account for all those differences that might even be happening in the same body of water.
All right, thank you. All right, our next talk this morning is Kalu Ogigwe from Clark University, mentored by Dr. Timothy Moore and sponsored by the Link Foundation. He's going to talk about validation of ocean color satellite products from Sentinel-2 over the Banana River Aeronaut OC site.
Thank you. My name is Carlo Kigwe. I'm from Clark University, Massachusetts. And this summer, we had the opportunity of validating ocean color satellite product, especially Sentinel-2, over the Banana River Aeronaut OC site. Dr. Tim Moore was my mentor, and was sponsored by the Link Foundation. So the project was basically, remote sensing is the acquisition of information without making physical contact with the object. So ocean color sensors dictate spectral light field emerging from a target. And that will show you what the target is, seagrass, optics, particles, chemically dissolved matters or whatever. Advances in sensor and satellite technology have increased in spatial resolution, has also increased from 1 kilometer pixels to about 1 meter in the last 30 years. Useful for making repeated synoptic observation virtually all at aquatic systems such as the Indian River Lagoon, which is a case study. But 90% of light reflected is from the atmosphere, so they need to be atmospherically corrected to be able to be used for any further study. Remote sensing reflectance is the desired quantity that comes from light emerging from top layer of the water. RORS, as it's called, is then used to derive properties such as turbidity, phytoplankton, biomass in subsequent algorithms. You can see that it's Sentinel-2 that we use for this study. We have Sentinel-2A, 2B, and 2C. 2A was launched in 2015, 2B was launched in 2017, 2C was launched in 2024. But each of them will capture a certain part of the place you're studying. But when you have them getting the images together, you're going to have a collective form of imaging coming not from one individual satellite. And when you have that combined, you can have a revisit time of 2 to 3 days instead of 5 to 10 days combined. This is the site, which is the Banana River in the Indian River Lagoon that we studied. Each S2 carries the multispectral image radiometer. And this has a spatial resolution of 10 meters. Spatial resolution has 5 channels, and those channels are 443, 490, 560, 665, and 705, and they are in nanometers wavelength. But this motivation for this is that how accurate is the satellite Sentinel over the Indian River Lagoon? This hasn't been confirmed by any research. So atmospheric correction remains a key challenge, particularly in an optical complex water system like the Indian River Lagoon. These projects seek to validate satellite-derived remote sensing reflectance product using field-based measurement. You have the Sea Prism radiometer, and it's located at the Banana River. There are about 30 of them operational, and Harbour Branch is controlling 2, one in Lake Okeechobee, one in Indian River Lagoon. And these validation efforts are crucial for refining water quality monitoring tools and improving atmospheric correction algorithm under various coastal conditions. What you see on the right is what you are going to get when you do a matchup of those satellite images against the Sea Prism data to confirm the performance. Before atmospheric correction, this is what a true color image of the Banana River will look like. This is Sentinel image, 10-meter resolution. After atmospheric correction, this is what it will look like, showing you intensity of the remote sensing reflectance is at 560 nanometer. This is the NASA Sea Prism Aeronite site. Let me play the video. Atmospheric correction is crucial for satellite studies. All satellite and sensors requires validation so that they can be used. So this Sea Prism is an automated robotic radiometer. And every 20 minutes, it takes sequence of water and sky measurements. And this will be used to validate the satellite measurements. Hopefully, when it's taking this, a satellite passes. So we're in the project, we did a matchup, trying to match up the satellite capturing with the recording from the NASA Sea Prism data. And this is available on the NASA website every 1 hour after processing. So you can use that to validate the satellite images. The research question is simple. How accurate are Sentinel-2 derived products after atmospheric correction? We use 2 schemes, the Acolyte scheme and the Radcore. And after that, does Radcore provide a significant improvement in atmospheric correction over acolyte in areas with strong stray light influence, areas like the Banana River, which is a coastline. Then these satellite-derived images we used was from 2023 to 2024, and the Institute data from the NASA Aeronet OCC Prism radiometer was the same. So environmental variable were also used from the Indian River Lagoon Observation Network, data on turbidity, wind speed, and chlorophyll air. So RADCOR, like I said, is a variation of the acolyte atmospheric correction scheme. Both were developed by the Royal Belgian Institute of Natural Sciences. All data and software are publicly available. So Sentinel-2 images was processed using acolyte, then we processed them again using RADCOR atmospheric correction. And after that, we used the C-Prism data to validate them. And this happened within a satellite overpass of 3 hours. So you have to match them, what we call match up, and compile them for statistical analysis. Additional environmental data, like I said, were collected from the buoy to contextualize the atmospheric and water conditioning after matchup. So this is the first result we get. You see the green light is the sea prism, the red is the acolyte, the blue is acolyte, red is RADCOR. So you could see the different matchups, and you could also see that different relationship between the three sensors, if I may use that word, after atmospheric correction. Then overall spectral shapes are similar. They all peak at 560 nanometer, which is the green, where the green dominates. In general, satellite 2 after atmospheric correction is higher than C-Prism, which is the grand truth, which you used to validate it. Then the next thing we see is that C-Prism versus RADCOR, you'll see that the Sentinel-2A and 2B showed similar results. Like I told you, that are sensors of 2A. You see A is in blue, sensor A, sensor B is in red. And you could also see that RADCOR on the left and acolyte in the next slide shows similar distribution. But RADCOR slightly better than acolyte. That's what our validations show for now. Error is very high for the satellite images, especially in the blue wavelength. This could be because of the sensor. It could be because of the atmospheric correction algorithm. It could also be because of seasonal influence, dry or wet season. Then you will see the error graph shown there and the bias also. And this is acolyte versus C-Prism, and you see that colored by month. Then if you go, what I told you about RADCOR and acolyte, they have similar distribution in the graph. But when you move further, you see that turbidity varies in the Banana River with wind speed. And wind speed is a big factor in controlling sediment resuspension. The C-Prism is affected by these events. For instance, you see in the graph that there was this Hurricane Milton that hit, and you see turbidity is very high, wind speed is also very high. On the left side, where you have shaded, is where you have turbidity is very high, while on the unshaded side, turbidity is very low. That's also shown in the ROS 560, which is the C-Prism. C-Prism is affected by these events, which are periodic and may last for several days. This is 2 weeks interval after the hurricane. Turbidity is a measure of particles, and is associated with higher ROS, especially in the green light. Then when you move further, the preliminary conclusions we have for this is that C-Prism and Sentinel-2 ROS data showed consistent spectral shapes and peak ROS value near the 560 nanometer, indicating an agreement in detecting major water color features. Acolyte and RADCOR generally track each other across the spectrum with minor differences in magnitude, especially at the shorter and longer wavelength. Sentinel-2 measurement after atmospheric correction overestimates remote sensing reflectance, especially at the blue wave light. Turbidity is an important indicator a determinant of RORS, and is also inflamed by wind events in the Banana River. RORS error in the satellite data will propagate into water quality products such as chlorophyll concentration, indicating high error to be expected. What are we going to do? Additional testing with additional atmospheric correction schemes will be used to validate this. We're going to use CEDAS, which is from NASA, and all that. There are dozens of atmospheric correctional schemes that is available. We only use 2 in this study, and we process about 200 images. And so what are the next steps? We take other atmospheric correction algorithms and additional satellite sensors. We broaden validation to include sites probably like Lake Okeechobee when we are done with this IRL. And we look at satellite platforms like Landsat 8 and 9, and probably MODIS, and develop better practices for statewide and inland, statewide based quality assessment in Florida. We're going to look at, present it probably at an ocean science conference in Germany and Glasgow when we are done. These are the references cited and used for this project. Then I want to thank Dr. Mo for his mentorship and guidance throughout this project, and the research lab in the Mo lab, especially Dr. Zach Wistock and EPA. Appreciation is also extended to the Harbor Branch Oceanographic Institute and the Aeronet OC program for providing data access and support. The funding provided by Lake Foundation was invaluable in analyzing this field work. Thank you.
We have time for a question.
Yeah, very nice work you've done there. Just very quick question. Do you think this analysis you did, can that be extended to some other remote sensing systems, for example, CubeSat, like the data from Planet Lab, they also have the multispectral sensors, might be, have higher spatial resolution?
Yes, it can. That's what I said. One of the things we try is we use something like MODIS or other sensor to validate. It can be extended, yes, because ability to validate what the satellite captures with what is field measurement will give us better understanding of the aquatic ecosystem, and it can apply to any kind of study: algal blooms, phytophans, seagrass mapping, and whatever.
Why do you think it's so bad in the blue?
Okay,
Just a quick one.
not sure what it is. We are still investigating that, but I'm thinking of sensor. I'm thinking also the atmospheric correction that was used. That's why we want to extend it to other atmospheric correction to see what is actually, whether it's sensor or it's seasonal or whatever. Yeah.
Cool. Thank you. All right, our next presentation is Andrew Fricano from the University of Rhode Island. He was mentored by Dr. Mike Twardowski and myself, sponsored by the Maryland Link Internship Endowment. He's going to talk about CASTLE, which is the Computer Analysis of Simulated Trace Bioluminescence Events.
Good morning, ladies and gentlemen. For those who do not know me, my name is Andrew Fricano. For those who do know me, my name is still Andrew Fricano. We got 12 minutes. I will be talking about CASTLE, which I am proud to present. As Dr. Wayne Slade gracefully introduced me, CASTLE is a computer analysis of stimulated trace bioluminescent events. So a little background about bioluminescence in general. Bioluminescence is the release of light through organisms in the sea and ocean. It's very common within dinoflagellates. It's triggered mostly by mechanical stimulation. This includes but not limited to waves, obstacles moving through the water like your hand, like a manatee or a fish, and something along those lines. It's typically followed by a very sharp rise by an exponential decline over a period of time depending on how sharp the rise was and what organism it was. So some motivation for CASTLE was the original goal is to automate detection, track, analyze flash events using an AI. To start this, we have to understand bioluminescence behavior like via flash detections. Many organisms will emit bioluminescence in response to intense mechanical stimulation, and manual video annotation is slow, inconsistent, and not really scalable because you're not going to have 200 people be working on one 4-hour video. It's a waste of time, it's a waste of resources, and it's just not worth the time. So the instrument we use to capture bioluminescence is called taquito. This is the field version of taquito right there. We have one underwater camera on this, one seabird pump, and one acrylic tube. Over here, we have the lab setup for taquito. It contains the same seabird pump, the same acrylic tube, the same Sony camera, which is the same one in the underwater camera, but we now add an Orca camera, which helps us get more data. Those are the two differences between them, and taquito stands for a tactical quantitative impeller to biooptics. So going into deeply what CASTLE is a CNN with a neural network. So what is a neural network? A neural network is like connects neurons. So we have neurons in our brain. It's how we think. It's how we reason thought. The CNN and neural network works together in that same process. It makes it so it's an automated thinking kind of like AI. And the CNN stands for a convoluted neural network and is built specifically for image and video processing. CNNs typically will go through and extract features like images, blobs, colors, shapes, edges, whatever is useful and whatever you've trained it to go through and do. So I'm now going to attempt to explain how a CNN works in 4 very simple steps. And to start this, we need an image, like a race car. How convenient. So for this image, I'm going to now look for the bolts on the front tire, that tire. So to do this, we're going to go with what's known as the convolution layer. This looks for the bolt very closely by scanning the tire and the overall image very closely. It goes through literally pixel by pixel. It'll go boom, 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 so it'll find something it might like. Once it finds something it likes, it's going to go down to what's known as the pooling layer. This will keep the clearest bolt-shaped objects, and once again, this is found by using shapes, edges, color, whatever is necessary that you have trained it to go through and find. So in this case, I've naturally put the best one right in the middle, and this will then go down, it's going to be thinking, it goes down to the fully connected layer. This decides whether or not it is a bolt. And so if you have something that's close to it, like on the edge of the tire, you see that little black outline there, that is something it would probably consider at first. This could be something that's worth going on to further processing to look. But then as it gets down, it's going to think to itself, no way, this is wrong. We're not going to do this. And it's going to go back. It's going to keep looking for a couple others, like the bolt center on the center of the tire. It's going to then think to itself, hey, we got something that fits the right shape, fits the right color. This is effectively a bolt. We can now process our data and process the bolts. So the CASTLE pipeline, we got a video right here. This is there's a mouse. This is the video of lab data, which was recorded in early June, and you'll see them going across the screen. This is without any video annotation. There they are. Beautiful. And CASTLE, once again, in case you forgot, stands for Computer Analysis of Stimulated Trace Bioluminescent Events. So the CASTLE general pipeline in 5 simple steps is it goes through video preprocessing, like that original video I have up there is what we call that. Then it goes into CNN flash-based detection, which goes back to what I just said a previous second ago. This is heavily about its convolution layer. Then we go into the motion tracking, brightness filtering, and output generation, which we will talk about in the next couple of slides. All these steps work together to isolate, track, and summarize valid flash events, or VFEs. Right? So that's enough of that video. We're going to now watch this video down here showing them with tracking. Now you might ask yourself, Andrew, why can I see this one so much better than the other one? Glad you've asked that. Let's wait for a nice big blob to go through. Like right there. Absolutely beautiful. Nobody asks yourself, why beautiful? So it's because we put boxes around this. And this is how I know and how we can see what the CNN sees and what it is processing and analyzing. So the video and image processing, the field and lab videos are converted to consistent frame sequences using FFmpeg. They ensure it's a 60 frames per second standard resolution and a standard format. I'm not trying to throw any tricks at this AI. It needs to not be looking the other way while we throw a kickball in its face, right? It needs to be ready to catch it, analyze it, and go. So this is what helps with the pre-processing, makes them ready. So that box around there is what it is specifically trained to look at it for. It is trained to go dark, 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 light. It basically thinks to itself, nothing, nothing, nothing. Oh, we got something. Here we go. We're doing great, right? So down here in the very bottom, you'll see this is 3 samples which I have taken from my data set. I have a positive sample of just a general noise and a negative sample. So you can see the very clear differences of how I was going back with the brightness threshold. You see we have a very bright, intense light compared to a noise, which is kind of just a camera filter and of it being at a high ISO, 409,400. So it's just a very high ISO. Some of that times that will come through and just be there. So we have to make sure it doesn't track that because that would lead to inaccurate results. It would lead to much more human processing and filtering than is what was initially designed to do. So this makes sure that we go through and we don't have to worry about that. On top of that, now we go into our flash-based detection. Like I said previously, each frame is passed through the trained CNN. It locates the organism in the blue channel and only the blue channel. You might be asking yourself, why the blue channel? Glad you asked. It's because these organisms typically flash in blue light, so we don't need to process the red and green channel. That would just overall slow down the pipeline. It would slow down the process, wasting time and resources, and makes it just more long and more lengthy. So once again, we're going to disregard the noise and background light like I showed previously, and the flashes must meet the direction set by the user, which is done by the user clicking on one side of the screen and clicking on the other. So everything going from like left to right of the screen is perfect, or however you set it to be. So if something goes right to left, we can immediately ignore it, even if it fits within our region of interest defined by the user. Once again, we go back to our brightness threshold. This is very important because, like that noise, such a high ISO, we're going to have some background noise of just general spark traces of light. It could be from other bioluminescence that somehow bounced off a wall or something in the background. It could be stars if we're out in the field or something, or a light, just car light. So we try to ignore that. And we also have to follow our size and shape criteria, like I showed within our positive sample. So right here we have a video of one organism, same organism. This is looped 9 times, going through at half speed, so we can see how it goes through and we can see how it tracks it. Beautiful, right? On top of that, we do go for and we find the velocity of the organism. We can do this because I know my flow rate of my tube, I know how long my tube is, I know how far my distance is, so it's just a simple distance divided by time calculation to find my velocity. And the velocity for the tube going through, the tube is about 1.6 meters per second, or a flow rate of 0.21 liters per second. So we can do this. I put a 50% error bound on this, so if it is going to be traveling at a minimum speed of 0.8 meters per second for in the right direction, within the region of interest, follow the brightness threshold, and be in the correct station type for it to be considered a positive result and for it to be processed. So, onto the motion tracking. After a flash is detected, positively identified, motion tracking follows the same object across the frame. Once again, through centroid direction and directional filtering. The tracks must be within the user-defined region of interest. Once again, exhibit a decaying brightness because, like I said in the very beginning, these are going to follow a sharp spike followed by an exponential decline. And then once again, be within the user-defined region of interest. So right there is what's known as a stack projection, a Z projection. This does, it takes frames from every frame from, like, let's say 68 to 102. It's going to put them all on top of each other like a big sandwich, and that's to get your output. Those little boxes over there are just overwrites from my video after tracking because the process goes through, it finds it, writes on the video, and then it goes back through and runs it again. So those boxes are just the overwrite. They are not part of the organism. The organism is the nice, beautiful little streak we have running through there. So outputs and results. After it's done running, you'll get a .avi clip with a couple frames before and after each organism individually. You will also get an entire video sequence with the entire organism, which I've showed both of those videos. You get a brightness versus time plot, a light decay curve, and a luminescence equation down there at the bottom is used to help calculate a lot of these graphs. This luminescence equation takes digital data from an image to a physical unit light level that we have calibrated light level for these cameras. Right here on the graph this is what's known as a luminescence versus time graph. So each one of those blue lines shows where Castle successfully found and identified a flash of bioluminescence. So there are 12 lines there, which is very important because if we come over here, down here on the bottom, we see we got how many were used to generate my histogram right there, total plankton clips saved, and total plankton tracks used. Oh my gosh, all the same number. We did something right. It worked beautifully. It worked perfectly. That is what we were looking for. So right over here at the top is our histogram. This shows you your average length of each flash. We got all sorts of ranges, with the majority being for this specific clip being from 0.2 seconds to 0.3 seconds. Now the average flash for the organism which I use to process this should be about 500 to 600 milliseconds, or about 0.5 to 0.6. So there's a little discrepancy. It could be because the pump is too fast. It could be my Castle's not picking up quickly enough. A couple of discrepancies here or there. So this is where it needs further testing, further improvement. So right over there, once again, we have the output folders. You can see all of the planktons have their individual clipped data within them, and with the overall data for the general process as we go along. Some future directions for this: we need to retrain and add more data. Currently, I've got about 100 pictures within each of the subsets, the noise, the background, and the positive. For a good CNN, it needs to have about 1,000 in each of those, so we got about 2,700 more images to go through and process, so that's going to be a fun weekend. I would like to add a GUI to make it more user-friendly, so I can send it off to some colleagues or my mentors, and they can just do it. They don't need to have me walk them through how to train it, like how to do it, so they can just have it and it's ready to go and it's done. Once again, I'd like to continue tuning so we can find all these flashes. We can help by some of my graphs, make them look more pretty, help them be more pretty and be more accurate. On top of that, I would like to add a potential adaptation for real-time analysis. So as we put Taquito in the field or in the lab, we can see it going through and we can analyze the data right away. We can make adjustments to the tube or to the pump as necessary as we are doing it for real time. I would like to give a special thank you to Dr. Mike Trusky, Dr. Wayne Slade, and Dr. Ed Malecki for being my mentors on this project. I'd also like to thank the Maryland Link Internship Endowment for providing me with this opportunity. Thank you. Time for questions?
Okay. So quick question about how you get your training data. Do you manually label the data or using some tools for the labeling?
So in case you didn't hear the question is about the training data, how I get it. Yes, I do manually go in and by each individual video I want to process, I'll go in like some just general videos. I'll just go and I'll just find, say, positive, negative, false, and just go in and do that till as long as I feel it's appropriate for me to get a more accurate data set. So in this case, I went for 100 and went off of there.
So did you consider maybe, remember we talked about using Make Sense AI? Would
Yes.
that be some tools?
That is definitely something I'm looking into to go on for the future side of this to make it more accurate and make it a little more machine learning so it can learn as it goes instead of me having to manually tell this is a flash versus everything else for the training data.
Great work.
Thank you.
Do you have multiple flashes from the same organism as it's traveling through the tube?
Yes, you can, and we did see that throughout the tube sometimes. That's known as a secondary flash, and creates like a sharp spine with like a little more bunny hop curve at the towards the end, and that's secondary flash or a death glow potentially if it's dead. So it does happen, yes.
So how do you account for that when you're counting how many organisms?
So I'm glad you asked that. So what it does, it'll follow the same organism through, and say it drops off for 2 frames. There is a buffer of 5 frames on either side. So if it sees the same one within the same general trajectory from like right here, and then it cuts off right here, and it comes back right here, it will link them together so it knows it's the same one still.
I'm curious. You did a great work, but I'm curious which convolutional neural network did you use? Did you train it by yourself?
Yes.
And if you did, which activation function did you apply? Or if you didn't and you used already a trained model?
No, I trained it by myself, and I went through manually training it. I went through the MATLAB CNN to help with that function and apply it.
Thanks.
Thank you.
All right, closing out today, this morning's session, Catherine Garrison from the University of California, Davis, mentors Dr. Michael McCoy, sponsored by the Link Foundation, and she's going to talk about assessing the utility of the invariability area relationship for forecasting regime shifts in the IRL.
Hi everyone, I'm Catherine, and this summer I had the privilege of joining Dr. Michael McCoy's lab here at Harbor Branch. And today I'm going to be talking a little bit about my project, which is assessing the utility of the invariability area relationship for forecasting regime shifts in the Indian River Lagoon. So for a little bit of background, the Indian River Lagoon, or the IRL, is a really important resource for Florida for many reasons. Namely, it's a biodiversity hotspot as it's home to over 9,000 species of organisms. It's also economically essential as it contributes about $7.6 billion annually to the economy. It also supports industries like aerospace, marine activities, tourism and recreation, as well as resource management. And lastly, it provides food, jobs, and housing to about 1.5 million people. So it's a hugely important resource. Because of its importance, much effort has gone into monitoring the health of it, including the benthic monitoring program, which is headed by Dr. Holly Sweat at the Smithsonian Marine Station in Fort Pierce. And what this program does is it looks at these 15 sites shown to the right, and they monitor at those sites quarterly. And this has been an ongoing project since the February of 2005. So the project collects data on the benthic and fauna communities as well as the associated environmental conditions. It's a massive data set and it's still expanding. But to date, they have collected around 3,420 sediment grabs, which have resulted in around 1,187 taxa spanning 16 phyla. And overall, that's yielded over 63,000 entries. So again, it's a really huge data set, making it highly informative and a great indicator of ecosystem health. Now to backtrack a little bit, when I say benthic infauna, benthic refers to the bottom of the sea floor, and infauna refers to those organisms that live within the sediment. So think tube worms, clams, things like that. The reason benthic infauna are chosen for this monitoring effort is because their short lifespans and their relatively limited mobility means that they respond rapidly to environmental disturbances. This makes them highly indicative of ecosystem health and restoration success. However, despite our access to this really expansive and informative data set, we've still seen incredible declines in ecological conditions in the Indian River Lagoon in recent years. So on the left here, we have this plot that shows the total seagrass coverage in the Indian River Lagoon. And if we follow this red line here in the middle, that kind of shows the averages. So we can see it kind of hovers around this stable equilibrium leading up till around 2011 when you see this dramatic decline. And then it kind of restabilizes, and then you have another decline around 2015. So these events coincide with the emergence of huge phytoplankton blooms, which, as we've learned earlier, can contribute to harmful algal blooms. Now, this shift has been associated, or it's been statistically shown that this shift is characterized as a regime shift. And an ecological regime shift just means that the ecosystem switches dramatically from one stable equilibrium to another totally different stable equilibrium in a very short amount of time. So in the Indian River Lagoon, this shift was characterized by a predominantly seagrass-dominated system to a more mudflat and algal bloom-dominated system. So in this graph to the right, we see we see that we have a graph of phytoplankton. So we see the relative abundance of phytoplankton is pretty low leading up till, again, around 2011 when this regime shift occurred, and you can see the spikes in the phytoplankton. And again, there are more spikes occurring around 2015 when a similar event occurred. So that brings me to my project. So I wanted to look at the invariability area relationship as a predictor for these regime shifts. So the invariability area relationship is just a new mathematical tool that allows us to assess ecosystem stability over different scales. So this plot is on a log-log scale, and we have invariability on the y-axis and area on the x-axis. So what it's showing is the invariability, which is essentially a way to measure the stability of an ecosystem as we change scales. Moving from left to right, we're going from a small scale to a larger scale, and we have this expected relationship which creates this curve. Now, the way in which we can use this tool to predict regime shifts is shown here. So this is a series of IARs stacked on top of one another, and they represent IARs for the same ecosystem under different ecological conditions or environmental conditions. So starting with that top line, the blue one, we see a very small perturbation to the system. And then as we move downward, we see how the shape of that curve changes as the perturbation grows in magnitude. And so we can expect the changes to look somewhat like this as a system approaches a regime shift. And there are three main metrics we can use to actually quantitatively and analyze how the shape is changing. Now, those metrics are first local invariability, which I'll talk a little bit more about in a minute, and the initial slope of the curve and the final slope of the curve. And the last thing we need to understand is how to quantify this relationship. So here's the governing equation for the invariability area relationship. We see in the denominator there that CV squared term. That represents the coefficient of variation. That's just a statistical metric that allows us to understand the amount of variation within our data. And it's essentially a measure of variability. So when we take the inverse of the variability, that's how we get invariability. Another way to understand this is to look at this right-hand side of the equation. Starting with that I sub 1 term, that represents local invariability, which again is one of those metrics that we want to analyze to be able to understand the changes in the curve. Local invariability is basically this. So imagine we start with just one site and we calculate the invariability at only that site. Then we go through and do the same thing independently for every single other site. Once we have all of those invariabilities, if we take the average, that's local invariability. Now, it manifests itself as the intercepts in our IARs, and that's because since we have area on the x-axis, the farthest left we go is the smallest scale we're looking at. So in our case, the smallest scale is one individual site, which is why the intercept is local invariability. And lastly, we need to understand that rho term in the denominator, which represents average temporal correlation as a function of distance. Basically, all that means is that in a given area, it's the average correlation between every single pair of sites. So what we expected to see, well, we knew that these regime shifts occurred in 2011 and a similar event again around 2015. So we were expecting to see this represented in our data through those 3 metrics that I talked about earlier: local invariability, initial slope, and final slope. So our first step was to construct a distance matrix and understand how far apart our sites were. What we did to do this was we essentially drew out a route that connected all of these sites within the waterways, and we calculated the distances using that. That yielded this distance matrix, which essentially shows how far each site is from every other site. From there, we were able to start constructing our invariability area relationship curves. So the way we went about that is we started with the data in our set from 2006 to 2008, and we looked at one site to begin with. So let's say it's this one. We would calculate the invariability just at that site. And by the way, we used the metric of total abundance. So then, after we had that local invariability, we would aggregate an additional site and just sum the total abundances between those sites, and then recalculate the invariability. We would do that again by adding another site, calculating the invariability, and we continued to do this one site at a time until we had encompassed the entire area we were looking at. Once we had that, we would repeat this whole process again, beginning with each of the other sites. And that, once we had all of that done, we would take the averages of the invariabilities at each specific scale. So looking at the average of the invariabilities of every 2 groups, 3 groups, etc. And that would produce a curve, a singular curve representative of that subset of years that we were looking at, 2006 to 2008. Then we would repeat that entire thing with an additional year of data. So now 2006 to 2009, yielding another curve. And we kept doing that until we had gone through our entire data up through 2021. So that yielded a series of curves, which are shown here. So we have invariability on the y-axis and then spatial aggregation along the x. Spatial aggregation just means the number of sites, so the scale that we're looking at. We can see that the local invariability is at that when we're looking at only one site. So every curve corresponds to a certain time period. So for example, that top one is the 2006 to 2008 that we began with, and each color corresponds to the ending year. So you can see the top curve ends at 2008, and then as we go down, it kind of extends the timeline. So then we wanted to analyze the changes in this curve. Our first metric to look at was local invariability. And as a reminder, this is what we expected to see as our system approached the regime shift in 2011. And this is what our data showed. So again, I want to focus on that data to the left of the 2011 there. We do see a decrease, which was in alignment with our expectations. However, we expected it to kind of bottom out there and not continue to decrease, but what we see instead is this continual decline. Now, we think that that's attributed to the fact that we were incorporating more data as we lengthen the time, the number of years that we were looking at, which inherently adds variability and can change the intercept. But that is something that we want to go back and look more into. But for now, to remove that bias, we just divided it out to look at the relative IAR. So we have temporal invariability divided by that local invariability on the y-axis here. This one, I like it because I think it just allows us to kind of more easily examine the changes in that shape as without the influence of that local invariability. Then the next metrics we wanted to look at was the initial slope. And again, as a reminder, this is the kind of decline that we were expecting to see leading to up to the regime shift. And here's what we got from our data. So if you look to the left of that red arrow, you can see up here that decline, which is exactly what we expected to see, and then a strong drop at 2011 where it kind of stabilized at around a lower value for a while until around 2019 when it jumped back up again. And that later increase, it corresponds to what we know to be positive reactions from the environment to restoration efforts. So this closely aligned with our expectations. And the final metric we wanted to look at was final slope. And so again, that was the expectation. We wanted to see it, or we were expecting to see a decrease. And then this is what we got. Again, we do have that strong drop leading up to 2011. And then after that, we have a couple of years that aren't quite what we would have expected, which would be interesting to look into. But then it settles back down at the bottom and again shows evidence of that positive restoration. So all in all, we concluded that the IAR seems to be very effective in acting as this kind of tool. It successfully reflected the regime shifts that we know to have occurred in the IRL around 2011. And so we think that it shows promising potential for becoming a tool to monitor ecological stability and inform adaptive management practices as future data is collected. It's also very feasible as a metric because it only requires total abundance rather than any specific indicator species or difficult to obtain data. So I'd like to take a minute to thank everybody who was involved in making this project possible, namely my advisor, Dr. Mike McCoy, and the other McCoy lab members, as well as Dr. Holly Sweat from the Smithsonian Marine Station. I'd also like to thank Harbor Branch and the Link Foundation for sponsoring this internship. And then I just have some cool other things I got to do as a part of my internship. So this was a day out in the field, and then here's some other photos of me in the field in the lab. I got to catch some lizards. That was pretty cool. And then I also got to hang out with some other cool animals. So we had some spider crabs, some anemones, sea slugs, and some other kinds of lizards. And then here's some image citations, and now I'd like to open it up to questions.
Time for a couple of questions before we head out for a break.
What will be the future? Will they continue to do this work after your intern? Set this up so that it will continue?
Yeah, so the monitoring effort is still ongoing. And yeah, this was kind of a preliminary study just to see if this invariability area relationship really could accurately predict these regime shifts. But because of our promising results, we are hoping that this will continue to be a way that they use to evaluate the ongoing data.
You mentioned that you just use total abundance.
Mm-hmm.
Can you use this method on more specific variables, like some particular slug or
Yes.
or macroalgae?
Yes.
Like, what would that tell you differently than total abundance?
Definitely. That is a future direction we're hoping to take, just to look at both just specific species or taxa, and also there are other metrics like species richness. And I think that would give just a bit of a more focused view as opposed to an entire ecosystem view, but it could also, depending on the species you're looking at, provide some more insight to specific factors.
Perhaps could help you show that it is actually recovery efforts that are having an effect.
Mm-hmm. Yeah. So overall, it is a tool used to evaluate ecosystem stability. We specifically focused on predicting this regime shift because there aren't really tools that are currently in use to do that. I don't know a whole lot about the other restoration evaluation techniques that they use, but I think they do have a lot of other metrics that they currently use. But this does show that, like, it would be a promising possibility to use it for that too.
Thank you very much.
Thank you.
All right, that concludes this morning's session. This was fantastic. Let's give the whole set of speakers another round of applause. And I believe we have a break now until 10:50, and we'll see you then.
Hello, everyone. I'm Dr. Paredes. I'm a faculty member here at Harbor Branch, and I'm going to chair this second session called Marine Science and Sustainable Aquaculture. So our first speaker is going to be Sky Flegg, and mentored by Megan Davis and Robinson Basurto. Just a little bit of an intro on Sky. Sky, she graduated from the University of New Hampshire with her bachelor's in biology, and this fall she will be starting Marine Science and Oceanography Master program here at HBO. Good for you. And one memorable moment from her internship was participating in the Queen Kong Aquaculture Training Workshop in the Grand Bahama. Welcome.
Hi, everyone. My name is Sky Flegg, and I was an intern in the Queen Conch Lab this summer, working with Dr. Megan Davis, Robinson Basurto, and Amanda Matthews. My project was evaluating the growth of microalgae to diversify the Queen Conch veliger diet. Okay, so for those of you that aren't familiar, the Queen Conch is a marine gastropod. It's well known for its bright pink, beautiful shell, as well as for its meat. So if you've had conch fritters here in Florida, you probably had imported Queen Conch. It's the most significant molluscan fishery in the Caribbean region, and is important not only economically, but also ecologically and culturally. So the Queen Conch Lab focuses on aquaculture as a means for helping to restore this species because it has been overharvested in many areas. The process of aquaculture for the Queen Conch starts with collecting egg masses and processing these. Then veligers or larvae hatch in the hatchery, so you can see a video of them swimming around. They're able to swim because they hatch with these two lobes, so that's how they are in their pelagic stage. And then they're raised in the hatchery. This is an example of one of our mobile labs, which is now in Grand Bahama. We put these together here at Harbor Branch and then ship them to various nations in the Caribbean as we work with partner organizations to bring Queen Conch aquaculture to their communities. Veligers then metamorphose around 21 days, so you can see a competent veliger has these 6 elongated lobes. They're exposed to a metamorphic queue, and then they become a benthic snail. So at this point, they're juvenile, and they're raised in the hatchery until release. So these veligers are fed microalgae from post-hatch to pre-metamorphosis. This is an example of a veliger feeding. So you can see on its lobes, there are cilia, and it uses these cilia like a conveyor belt to pull in that microalgae, which are all the little dots, into its mouth. So the goal of my project was to identify a more diverse diet for the Queen Conch that produces high survival rates, shorter time to metamorphic competency, as literature has shown that veligers can reach competency in as little as 13 days with a more diverse diet, compared to our average of 21 days. And then more algae will provide more options for hatcheries because, as I mentioned, they're located in many different areas. So one algae species might grow great for one hatchery and then not so well for another. So if we have more species, then there's essentially more tools in the toolbox for these hatcheries. The objectives were to assess the growth of golden brown and green algae in culture, as well as the growth of diatom algae in culture. The current veliger microalgae diet consists of 2 golden brown algae, Caicos isochrysis and Tahitian isochrysis, which are CI and TI. These are fed throughout the veliger stage, and then Chaetoceros gracilis, a diatom, is introduced around day 16 later on in the veliger phase. In addition to those species, I worked with some experimental microalgae feed, including 2 green algae species, Tetrasalmus chewi and Nanochloropsis oculate, as well as Chaetoceros muelleri and Cyclotella nana, which are 2 diatoms. So how did I go about growing these algae here at Harbor Branch? You can see in this first photo is an incubator. In this incubator, I had both tube cultures and flask cultures. I'm going to focus mainly on the tube cultures. For these, I had 4 replicates of each species. And as I was going to inoculate these replicates, I would autoclave all of my materials the day before. So this included the tubes, caps, pipettes, as well as the filtered seawater, which is the basis of the media. I would then choose the new pure culture. So I did this by assessing any current replicates I had for some qualitative features, which I'll get into. But this way, I always carried on the best, cleanest culture that I had. And then I would prepare the media. So this included nutrients, which are part A and part B, which you can see are those brown and pink bottles. This includes trace minerals and metals that the algae need to grow, as well as silicates and vitamin C for the diatom media only. This is because diatoms have a silicate cell wall, so they need the silicates to grow that. And then once you add silicates to media, the pH goes up. So we add the vitamin C as a buffer to bring the pH back down to a normal range. Media is then added to the tubes or flasks and inoculated with the pure culture at a 10-to-1 dilution. And the algae is swirled twice a day, every day, in order to ensure that algal cells are staying in suspension in that media. For data collection, you can see in this first photo, I'm working with an open culture. Anytime I had an open culture, it was near an open flame. That reduced any potential contamination in the air that could get into the culture. I'm loading a slide called a hemocytometer, which is generally used to count blood cells, but it works really well for algae cells. You can see what that looks like while using the microscope in that second photo. So the first thing I would assess is shape, motility, and contamination. These are those qualitative factors that I mentioned I would use when assessing which replicate to use as my new pure culture. And then I would assess density. So in that hemocytometer slide, you can see that there's 25 total squares. I would count all of the cells within those highlighted 5 squares and then multiply that by 5 to get the total 25 squares. And that gave me a 0.001 milliliter sample. So then I multiplied that number by 10,000 to get my total cells per milliliter. And these counts were done on days 4 through 7, with day 0 being the day of inoculation. As for my results, the first thing I went through doing was getting my standard deviations. So this compared each of the 4 replicates within each species and essentially told me that all of the replicates for each species, apart from TC or Tetrasomus, which I'll get into, grew very similarly to one another. So I was able to average the results of those 4 replicates and then carry on working with that average number. For Tetrasomus chewi or TC, we can see that the standard deviations are pretty high. This is likely because the cell count remained very low throughout the study, and there was likely an increase in the technical and biological noise because of that low cell density. Before I get into my graphs, I wanted to show you what the algae growth curve looks like. So microalgae grows logarithmically, meaning it has this short lag phase and then exponential growth, a stationary phase or plateau, and then a culture crash. So we try and keep our algae in the exponential growth phase, where we harvest for both inoculation and feeding. So for our purposes, we want the exponential phase within the tubes to go through day 7. This is because in an operational hatchery setting, we want to only have to do new inoculations once a week. So if we can have that exponential phase go through day 7, we can then have a schedule in a hatchery of doing that once a week. So first up, we have the green and golden brown algae. In this first graph, you can see the mean tube culture density in cells per milliliter, which is also on the y-axis. On the x-axis, we have days 0 through 7, with that dashed line representing estimated growth, because again, my counts were days 4 through 7. We have on the other graph, mean specific growth rate in mu per day. And the way that this is read is on the x-axis, we have days 0 through 4, and then 4 to 5, 5 to 6, and so on. On the y-axis, we have mu per day. So if you're looking, for example, at TI or Tahitian isocrisis, it's that first dark blue bar. That's at about 0.28, meaning this culture grew 28% per day between days 0 and 8. So first we'll look at TI and CI, which are the dark blue and orange lines. So if you look at their density, we can see that continued to increase throughout the 7 days, which we would like to see. And then for the mean specific growth rate, when we're looking to see if it was growing exponentially, we want to see a stable growth rate throughout the 7 days, which for these species we did see with a little bit of fluctuation, but for the most part, it was very stable. So that indicates that these 2 species were growing exponentially throughout the 7 days. So they grew very well in these conditions. For Tetrasomus and Nanochloropsis, or TC and NO, we saw a little bit of variability for these species. So I'll focus on Nanochloropsis because you can see it a bit better. As I mentioned, TC had a very low cell count. For Nano, we can see that it grew through day 6 with an increase in density. And then if you look at the specific growth rate, we can see that it dropped quite a lot after day 4. So it's that light blue bar. So when So it dropped after day 4, indicating that it was starting to go into its stationary phase, despite what we were seeing with the density, because the density didn't drop until around day 6. So that's why it's so important to have these 2 pieces of information to give you the full story of what's happening with your culture, because if we were just looking at density, we might think it was growing exponentially through day 6, when in reality, it likely was starting to go into a stationary phase after day 4. For the diatoms, we have the same 2 pieces of information. So this is CG, CM, and CN. This is really great because you can actually see that plateau happen. Well, great for you guys, so you can see what I'm talking about. But the cultures will focus on CG and CM just because they grew very similarly as well. They increased in density throughout day 5, but the mean specific growth rate dropped quite a lot after day 4. So again, indicating that it was going to go into the stationary phase before we actually saw that stationary phase represented with the density. And then similarly for Cyclotella, we saw that happen after day 5 with the specific growth rate dropping and then the culture going into a stationary phase. So overall, I would recommend adjusting the nutrient levels to extend the exponential growth phase through day 7. We can do this through incrementally increasing the nutrient concentrations. So we'll slowly add more nutrients and then assess how the algae grows with increased nutrients. You don't want to add too much if you're thinking like, well, just throw a bunch in there and it'll grow great. If you add too much nutrients, then bacteria and protozoa can actually start to take a hold of your culture. So that's why we want to do it in increments. You can also track cell density and nutrient correlations. So this would look like doing cell counts and then taking actual water quality samples of your of each of your replicates and assessing that for nitrogen and phosphorus levels. This would be very labor intensive, but would give you more good insight. For further optimization, I would recommend adjusting intensity and photo periods for in terms of light for these species. So particularly with Tetrasalmus chewi, as I mentioned, it remained very low. So our hypothesis is that that species might be limited by more than just nutrients and will likely introduce photo periods. For salinity, we can also adjust that to optimal salinity ranges for these species. So this study was run at 33 parts per thousand, and each species has their own optimal ranges that we can adjust it to. Temperature can also be adjusted to optimal ranges based on thermal tolerances. So this study was run at 76 degrees Fahrenheit, which again can be altered. And inoculation density can also be changed. So if you recall, like Nanochloropsis, that light blue line had gone very high, but it was inoculated at a very high density. So we can also lower that to prevent early resource depletion as well as self-shading effects. In conclusion, all of the experimental algae species are viable to grow and feed to villagers. And as Fernando mentioned, I will be staying on here for my master's, and my thesis work will focus on further optimization of these species, as well as doing test feed trials to milk conch villagers, which is what we work with here in Florida instead of the queen conch, since those are protected. But milk conch are very closely related to queen conch. I would like to thank my mentors for this project, Dr. Megan Davis, Robinson Bezerto, and Amanda Matthews, as well as Dylan Gronley for his assistance. I'd like to thank the Ziegler Lab, Dr. Malcolm McNabb, and Richard Baptiste for their help with materials on this project, and of course the generous funders that made this project happen, the Link Foundation and Longer Tables. And thank you to all of you for being here. Any questions?
Questions?
Hi, great presentation.
Thank you.
Is there any sort of like preferred uptake for the queen conch villagers in terms of like which phytoplankton species? Do they have a preference between the different species?
So in the wild, they'll eat all of the species. I mean, for example, the current feeding schedule, we have those golden brown algae, which are smaller, and then Chaetoceros gracilis is a little bit larger, but we mainly introduce it like later on because it's very high in lipid content. So we use it essentially like to fatten them up for metamorphosis. But that's, I mean, there's more trials needed to kind of see what they'll eat most healthy algae, but yeah, just to see what makes them grow the best. Yeah, thank you.
Anybody else? Well, thank you very much.
Thank you.
Our next speaker is Nick Beswick-Sadel, and to quote Nick, he says, "I'm a rising senior at Wilmette University all the way in Salem, Oregon. My passion for marine science began when I was growing and propagating coral in aquaria from the Landlocked Boys, Idaho. In addition to enjoying game nights and trivia with other interns, I also got certified as a rescue diver this summer." Good.
Hello, everybody. My name is Nick Beswick-Seidel, and I'm a summer intern here at Harbor Branch, coming from my undergraduate at Willamette University. This summer, my project was working to establish a nutritional profile for 4 Florida native halophyte species for their use in aquafeeds. I was mentored by a host of people, including Megan Davis, Paul Wills, Fernando Paredes, Allison Chin, and Amanda Matthews. My internship was sponsored by the Link Foundation, and my research that I'm presenting to you today was sponsored by the Harbor Branch Oceanographic Institute Foundation through the license plate program, as well as the USDA Agricultural Research Services. So I want to start with you guys today by introducing what is a halophyte. Halophytes are terrestrial and aquatic sea vegetables, which can be grown in both soils or aquaculture systems. They have high salt tolerance, which means in addition to being able to be grown in a large variety of environments, they can also be irrigated with salt water or even grown in saline aquaculture systems. These 4 species, which I'm introducing to you today, Sueda linearis, Sesuvium portolacastrum, Salicornia bigelovi, and Baetis maritima are all Florida native halophytes. Due to their Due to their high tolerance to salt, they also end up being mineral rich, which can be a beneficial factor for aquafeeds. So I'm talking to you about aquaculture today and diversifying feed sources because since 1990, aquaculture has been growing rapidly, helping to take over what has not been accounted for by wild-caught fisheries. Now, in conjunction with this, a need for aquaculture resources to feed everything that you're growing is required. Now, today, that is primarily sourced from things like fish meal or products of industrial agriculture, such as wheat meal, corn, or even soy. For example, a diet being fed here to Florida pompano at Harbor Branch in a recent study was comprised of 50% fish meal, 15% soybean meal, and 14% wheat grain flour. So the goal and objectives of my project was to contribute the nutritional profile of these 4 Florida native halophyte species to a new FAU Harbor Branch aquafeed database, which is being pioneered by one of my mentors, Fernando Paredes. In doing this, I wanted to use proximate nutrient analysis to establish baseline compositional differences between wild and aquacultured halophytes. I was going to begin quantifying fatty acids for each of these species, again with both wild and aquacultured halophytes, as well as collect samples for further analyses, including mineral and amino acid analysis. As for the halophytes that were being grown here at Harbor Branch, they are being grown in what is called an integrated multi-trophic aquaculture system. Essentially, what was being done here is that fish waste and feed waste, in this case from the Atlantic croaker, is being recycled into other ingestible products such as Ovolactuca, a macroalgae, and our sea vegetables. You might hear me refer to this later as the IMTA system. As for harvesting, our wild halophytes were harvested from an Indian River Land Trust property in Vero Beach. And of course, the aquacultured ones were collected from here on Harbor Branch. Thank you, Jake and Ellie, for your help. After we collected those, they were divided into 3 tissue types. We had edible, inedible, and root tissues. After that, they were blended to homogenize the samples and placed into a drying oven where they dried at 39 degrees Celsius for 4 days. Here you can see the product of that drying on the bottom left of your screen, or bottom right of your screen, and then after this, they were blended once again to powder the samples. The methods for proximate analysis essentially followed 2 general train of thoughts. For method 1, you started with a sample of a known mass in a filter bag. Then you would place that filter bag through a solvent extraction, using something like petroleum ether to extract, say, crude fat from the sample. Then you would record a final mass in order to determine how much of that nutritional component in the sample you lost. Method 2 is similar. You start with a sample of a known weight. In this case, you're actually going to burn it, and from that, you're measuring something. In the case of calorimetry, that could be the heat emitted from your sample. In the case of protein analysis, that could be the amount of nitrogen. For the second part of my study, we are looking at fatty acid quantification. Essentially, you have your samples and you're extracting it with a variety of solvents, such as dichloromethane, evaporating it, and there's a few other steps there to separate your solid sample from the fatty acid particulates. One of the next presentations, presented by Jake Morris, will talk more in depth about this. These samples then go into a gas chromatography mass spectrometer, which is an instrument that can determine the molecular weight of samples, and we use that to determine what is actually there. I'll get into some of the results. For crude and on each of these introductory slides, I wanted to showcase some of the instruments which are being used to conduct these analyses. For each of these results portions, I'll display a graph something like this that is going to have your species on the bottom x-axis, the tissue type that I separated on the top, and then the y-axis is going to be the percent composition of something, in this case protein. All significant results will be highlighted using these asterisks, and aquaculture specimens will be circles, while the wild collected specimens will be triangles. Now I want to highlight here that our aquaculture specimens all had significantly higher levels of protein in their edible plant portions. Sometimes this was as high as twofold, giving us increases from around 10% to 17, 20, or even 25% crude protein. Now, for the rest of this presentation, I'm going to be focusing on Salicornia and Sueda, even though here you might see Symposium as having beneficial results for us. This is because in the context of aquaculture, you need to have something that you're collecting to feed the fish. Now, in our case, in this study, we had really extreme growth from Sueda, which I'm showing to you here on the left and right sides of this tank, whereas our Symposium is in that middle tank. Now, in other systems, and even our system in previous years, Symposium and Baetis have both been shown to grow rapidly, like Salicornia and Sueda. However, we did not see that in this year's study. For the inedible portion of our plants, we again saw increases in the amount of crude protein in the aquacultured samples compared to the wild samples, with the exception of Baetis. As for root, all 4 of our halophyte samples saw drastic increases, and statistically significant ones, in the amount of protein present in the root systems. As for crude fiber, we actually saw a lower amount of fiber present in our aquacultured samples, likely due to the aquatic environment they were grown in. This trend continued reversed for Salicornia, giving us more fiber in our inedible portions, however, maintained constant for Sueda. And then for roots, we saw that all of our wild samples had significantly higher amounts of fiber, likely due to the fact that they were grown in soils versus aquaculture rafts. As for caloric content, the results may not have been as extreme. However, for Salicornia and Sueda, we still saw that there was a significant increase in the amount of calories per gram in our aquaculture edible portions. This was similar for Salicornia in the inedible part. However, again, for Sueda, this inverted itself. For roots, in most cases that were significant, we saw a higher caloric content in the wild specimens, likely due to their dense woody nature when compared to the aquacultured ones. Now, there are 2 other important parts of proximate analysis, which are ash and crude fat. For the context of today's study, I'm not going to be talking about the results of these because in aquaculture feed formulation, the difference between 1.2% and 0.82% really isn't that great. And the same thing goes for crude fat, with wild coming in at an average of 1.28 and aquaculture at 1.03%. However, there are further analyses that could be done to make this significant for aquaculture feeds. For example, mineral analysis or fatty acid analysis to determine the composition of these 2 pieces. Now I want to introduce you to some of the other common ingredients in aquafeeds, which are whole wheat flour, cornmeal, and whole grain sorghum, which was actually used in a recent study with Florida Pompano at Harbor Branch. For protein, you'll see that our edible portions for both Sueda and Salicornia were much higher than those of the standards in aquaculture today, whole wheat flour and cornmeal, versus our inedible portions, which were similar to those of the standards today. Now, as for fiber, our edible portions, again, came in at a similar level to those of the standards, whereas our inedible portions had significantly higher levels of fiber, as high as 30.8%. Now, while fiber is indigestible in fish feeds, it does play other roles. Typically, a lot of fish feed producers have to add cellulose to their samples to give it volume or structure. In this case, if a producer knew that they needed to add fiber to a diet like cellulose, they could include these inedible portions to fill that role, eliminating the need to purchase another ingredient. As for fat, our results were similar for the edible portions to whole wheat flour, but lower than cornmeal and whole grain sorghum. However, for our inedible portions, they were undetectable from our methods that we used. As for caloric content, the edible portions were at or above that of cornmeal and whole grain sorghum, whereas the inedible portions were comparable to whole wheat flour. Now, I did mention fatty acids previously. We began getting some preliminary data for that, which I wanted to display to you here. It looked like a lot of our samples were very high in C16, LNA, and C22-2N6, which are all important precursors to fatty acids, essential fatty acids in fish diets. So this is exciting news. We're still trying to move into further analyses to see how they vary between our samples. As for my conclusion, we completed all three of my objectives by doing proximate analysis, beginning the fatty acid analysis process, and getting some preliminary results. And I've collected samples for that mineral analysis and amino acid analysis, which I mentioned previously. My mentors, Paul Wills and Fernando Paredes, will also be using this preliminary information to apply for the BARD Aquaculture and Desert Farming Program grant. In addition to this, Salicornia is being used as a feed for black soldier fly larvae, which is being analyzed for protein inclusion in feeds. What will be happening soon or in the near future? This will be a part of the first entry into the FAU Harbor Branch Aquafeed Ingredient Database, being pioneered by my mentor, Fernando Paredes, which will eventually be used to support development of on-site feed production at Harbor Branch catered towards small-scale aquaculture producers. Another interesting thing is that the goal of the IMTA system is to use every product that we can from the fish waste or plants that are being grown from it. So by finding a use for potentially the edible and inedible portions of the halophytes, we are helping to close that cycle by being able to include those into the fish feeds entering the system. Thank you all for being here today. I'd like to thank the Indian River Land Trust for assisting in the collection of my wild halophytes we studied today, as well as my expansive mentorship committee for their guidance throughout this summer. I'd also like to thank the other interns, especially those who helped me with my analysis and collection along the way. And thank everybody for the great game nights or trivia nights that we had, as well as the Link Foundation for funding my internship, the Harbor Branch Oceanographic Institute license plate program, and the USDA Agriculture Research Services for funding my project. Thank you.
Questions for Nick?
For development of future use of these halophytes, would you assume that people would be doing wild collection, or would you guys be providing seeds for these smaller quantities?
So the interesting thing about these halophytes, and what we are looking at, is to see if there was a difference between the aquacultured halophytes and the wild halophytes. And a protein content difference of around 10% to 20% is really significant, especially when you're looking at trying to remove as much fish meal from diets as you can. So from this, I personally would recommend aquacultured halophytes. However, further analysis could provide insight on which may better suit a purpose.
So why do you think the aquaculture halophytes have such a higher percentage of protein?
Yeah, so the IMTA system is generally characterized as a very nutrient-dense system. I should have included the numbers here, but off the top of my head, the nitrates in the system were around 30, compared to the really sandy soil where we are collecting our wild halophytes from up in Vero Beach. It's likely that our high nutrient environment was the IMTA system compared to that of the wild collected halophytes. And something that some people have talked about is doing further analyses at different sites that maybe were more aquatic or in different locale in order to determine if that's a constant thing or was unique to our site.
We have time for more.
One question. You think that not everybody has the access to get the water from that? Somebody want to do this in land, it's kind of replicable that you can do it or manipulate it?
You may be able to manipulate the nutrients. I mean, in conventional agriculture, there's fertilizers that can be done to do that. So it would certainly have to be researched, but there is potential for agriculture of these halophytes, especially because of their salt tolerance in deserts or places that may not have access to a lot of fresh water.
Nice. Thank you.
Thank you, Fernando.
Well, our next speaker is Ellen Gortz, and mentored by Dr. Marty Ritchie and Jenna Baggett. And Ellen Gortz is a rising senior of Old Dominion University during her time at HBOI. She gained hands-on experience in live feed nutrition research and learned how targeted enrichment can support larval fish development. This internship deepened her understanding of applied aquaculture and the critical role of rotifers in hatchery success. Go ahead.
Good morning. My name is Ellen Gortz, and I worked with Dr. Marty Ritchie and Jenna Baggett this summer to present the nutrient enrichment kinetics of Branchius rontamiferus, which is a study of tryptophan retention and uptake. Tryptophan's role isn't just nutritional. It also affects behavior. In larval fish, aggression and cannibalism can severely reduce survival. By increasing serotonin via dietary tryptophan, studies have shown behavioral improvements. Yet, most research overlooks how this nutrient gets into fish in the first place, and we want it through rotifers. Because rotifers can act as a delivery vehicle for tryptophan, but we don't fully understand how efficient it is to holding on to it. If tryptophan leaks out or degrades too quickly, fish may never benefit from it. This gap could undermine hatchery success despite their good intentions. The study focuses on the rotifer, not the fish, because they are the foundation of early larval diets. By better understanding tryptophan retention kinetics, hatcheries can improve timing and dosing strategies to deliver nutrients more effectively, ultimately supporting healthier fish populations and cost-effective productions. So this was our strategy. First, we looked into the amount, which varied from 0.05% to 0.1%, 0.15%, 0.2%, 0.3%, and 0.4%. We then used that percentage, as we saw was optimal, and moved to different retention or enrichment times. These varied from time point 0, which was immediately after harvesting, to 30 minutes into enrichment, 1 hour, 1.5 hours, 2 hours, and lastly 4 hours. After that, we decided on the most effective retention time and moved to metabolism. After enriching the rotifers for a constant amount of tryptophan and for a constant amount of enrichment time, we let them sit post-flush, varying hours, meaning we're moving from the enrichment, from 0 hours, which was immediately after rinsing, to 1 hour, 2 hours, 3 hours, and 4 hours, to see how fast the tryptophan will deplete. So this is the timeline that we used for each study. This timeline goes across 5 days. On the first day, we harvested the rotifers in the early mornings to determine how much volume of rotifers we needed to give each beaker. Then, we prepped the samples to enrich. Throughout all studies, the timer would start as soon as the first beaker of each replicate was topped off with salt water. Then, after the corresponding amount of times for that study, we sampled using a filter bag to flush and then transfer the rotifers into a sieve to fully ensure a full flush from that enrichment. We then transferred the rotifers into 15 ml Falcon tubes and freeze-dried them for 2 days straight to ensure a full freezing of each sample. After those 2 days were up, we prepped each sample for the assay kit to determine the tryptophan content, which took around 2 days. And then finally, we analyzed the data given from the microplate reader to analyze our next step. So let's break it down even farther. For each experimental unit, we consisted of 1 2-liter beaker. The volume of rotifers within each beaker varied depending on the morning harvest, though each beaker consistently contained a population of approximately 3 million rotifers. These rotifers were gently poured into the beaker, which was equipped with an air stone to maintain circulation. The tryptophan supplement was prepared by dissolving an appropriate amount of tryptophan in 1 liter of salt water and blending it for 3 minutes. To this solution, 0.692 grams of Ori-Green enrichment was added, followed by an additional 90 seconds of blending. The mixture was then allowed to settle for 5 minutes and then blended once more for 90 seconds to ensure a complete homogenization. These enriched solutions were then carefully poured into the rotifer-containing beaker to minimize stress or shock to these organisms. Finally, the remaining volume was brought up to 2 liters using filtered salt water. So I know I just gave a lot of information, so here's a visual guide of how we incorporated the supplement into the enrichment. By first weighing the corresponding amounts of enrichment in tryptophan, and then by blending in the corresponding order, and then enriching for the corresponding amount of time. Now to harvest. We first rinsed the rotifers using a filtered bag, and then they were passed through a fine 40 to 50 micron sieve to remove debris. Finally, we collected them into a labeled falcon tube and immediately placed them on dry ice and later on to a negative 20 degrees Celsius freezer to preserve their condition for sample prep. For all 3 of our studies, after a minimum of 24 hours at negative 20 degrees Celsius, samples were freeze-dried for 48 hours to remove moisture while minimizing contamination using Kim-wipes and parafilms acting as caps. Each freeze-dried sample was weighed out to 0.25 grams and homogenized in a freshly prepared assay buffer. After cooling and a second round of homogenization, we centrifuged the samples to extract the supernatant. Protein precipitation was done by using trichloric acid, and then followed by neutralization using sodium bicarbonate. These resulting clarified supernatants were stored at negative 20 degrees Celsius until analysis. To quantify tryptophan, we used the Cell Biolab's fluorometric assay kit. A standard curve was prepared fresh each day of the sample through serial dilution, starting from 100 micromoles down to 0 micromoles. Samples and standards were then loaded into duplicates, with each unknown sample split into 2 wells, one with tryptophan oxidase, known as plus TO, and one without, known as minus TO, to control for background fluorescence. After adding the appropriate reaction mixes and incubating the plate for 1 hour in a dark, at-room temperature, we then measured the fluorescence using the microplate reader. The difference between the plus TO and the minus TO wells allowed to accurate quantification of enzymatically converted tryptophan. As seen in the photo, the pink wells were where the plus TO reacted, whereas the minus TO did not. So here's what we found. We first start off with the relationship between the L-tryptophan concentration and the uptake percentages in rotifers. These graphs highlight the difference in R-squared values when the 0.2% concentration is excluded, suggesting the need for retesting to confirm the observed trend and ensure accurate optimization of nutrient enrichment. For study 1, as seen here, the 0.3% seemed to be the peak with being obtained with regards with how much was given. So we went with that concentration moving forward with this research. For study 2, focusing on the time versus retention of the tryptophan, we see this very nice climb as well as a drop. This supports that 2 hours was the most efficient time to use with regards to retention. In this post-flush metabolic time-based study, we saw a gradual increase of tryptophan concentration over time. Yet there was a noticeable spike in the tryptophan retention at the 4-hour mark, contradicting the time points prior. This could be due to consistent absorption in tryptophan over time. However, the sharp increase also raises concerns about potential sampling inconsistencies. Because of this, additional replicates or retesting may be necessary to confirm the accuracy of this result. So let's put this all together. We found that rotifers took up most tryptophan at 0.3% concentration with peak absorption happening around the 2-hour mark. This suggests that the 0.3% of 2 hours may be the best concentration for efficient enrichment. There were some odd results at the 0.2% and a metabolic 4-hour time point, so those need to be repeated to rule out sampling and biological variability. These findings are a step forward to making enrichment protocols more consistent and cost-effective with regards to tryptophan supplementation. By improving how we deliver nutrients like tryptophan, we can turn rotifers to more than just feed. They become active carriers to targeted nutrition for larval fish, supporting sustainable aquaculture. So here are our future implications. Looking ahead, future research could explore how feeding larval fish with tryptophan-enriched rotifers influences their behaviors and physical development. This could help us better understand how nutritional enrichment impacts early fish growth and welfare. Additionally, this study lays out the groundwork for supplementation for other live feeds, such as Artemia, with tryptophan or other similar compounds to improve aquaculture compounds. I want to sincerely thank Dr. Marty Ritchie for his mentorship and guidance, as well as Jenna Baggett for her continued support and feedback. Maddie Wheeler provided valuable insights, and Allison Chin was a huge help in the lab. Christopher Robinson assisted with setting up the experiments, and I'm also really grateful for my fellow interns with their collaboration throughout the summer. This research was made possible by the facilities of HBOY and generous support from the Link Foundation, which allowed me to live financially stable during this internship, and funding from the USDA. Thank you. Do we have any questions? No?
I have a question for you.
Yeah.
What do you think if this cannibalism behavior will still remain even if you feed them a lot?
Yeah. So obviously it's my hope that if we feed them enough tryptophan, the fish themselves will eventually calm down. But I mean, that's all up for future research. We really don't know because this is a full first thing. So it could be really interesting.
Great presentation. That was a lot of very detailed work.
Thank you.
How did you feel about all of that? Keeping track of everything.
Oh, I loved it. Loved it, loved it, loved it. It was also most promising to get really promising results that align with what our protocol is here for enrichment here is today. So it was really nice to see. Yes. Oh, yeah, for sure. Well, I mean, tryptophan comes from eating turkey on Thanksgiving. So, I mean, like, we can totally use it for ourselves if we're feeling a little too crazy. Yes, thank you.
We're going to wait a couple of minutes to keep a track. Okay, are you ready? So our next speaker is Jake Morris, and to quote Jake, he says, "When I started this experience, I knew nothing about aquaculture. But since then, I've learned to use key instruments, gain insight into fish feed, and earn my fish handling certification. Working alongside some of the most brilliant minds in the field has been incredibly rewarding, and I've loved exploring all the unique places in Florida." Good.
All right. Hello, everybody. I'm Jake, and this summer I've worked with Fernanda Paredes, Allison Chin, Ben Penitz, and Paul Wills in researching differences in fatty acid composition of naturally and artificially spawned eggs in California yellowtail. I would first like to start with some background information. First, why are we studying the California yellowtail? They can grow very rapidly, anywhere from 7 to 11 pounds in about 12 to 18 months. They also have become increasingly valuable, ranging anywhere from $11 to $23 per pound due to their high-quality flesh. Also, sustainable farming of California yellowtail can reduce pressure on other, more overfished populations, such as the bluefin tuna. And what are the importance of fatty acids? When the embryo is within the egg, it receives fatty acids from the broodstock, which is determined by that broodstock diet. So what goes into the broodstock ends up in the embryo in the egg. While it's in the egg, this is all the embryo has to work with in order to grow, develop, and hatch. So some uses while it's in the egg is to metabolize it into energy and determine its membrane integrity. As a result, fatty acid composition has become an important part in determining egg quality. The current method for finding fatty acid concentrations is gas chromatography mass spectrometry, otherwise known as GCMS. The problem with GCMS is that it takes a long time to analyze samples, anywhere from 4 days to about a week per sample. It's also not very portable, it's very large and pretty heavy, and it's rather expensive, as a unit can cost about $250,000. In contrast, Raman spectroscopy has the potential to deliver immediate, on-site results, enabling aquaculture producers to assess egg quality within seconds. By integrating fatty acid composition data into a predictive model, the study seeks to further bolster and validate research for a more accessible and efficient approach to egg quality assessment. Along with this, it's significantly cheaper at about a third of the price, being about $75,000. So let's jot down some objectives going into this research. First, we're going to determine differences in the fatty acid profiles of naturally and artificially spawned eggs, and we're going to improve a rapid on-farm fatty acid predictive model using Raman technology. Our hypothesis is that the fatty acid composition of the eggs that respond artificially will not be significantly different than those that respond naturally. Now let's look at the experimental design and get a better idea of the variables during this research. First, you have the broodstock spawning method, natural and artificial. Artificial meaning that the spawns were artificially induced by controlled environmental cues, in our case light and temperature. You have the tanks in which they were stored, the periods in which they had their spawns, and the number of spawns from each of those time periods. Let's dive even deeper into the difference between natural and artificial. Both groups were fed the same cut-bait diet composed of sardines and squid, kept in the same size tanks, and had the same standard conditions. The only changes were once again environmental cues, in an attempt to replicate summer conditions during the winter months in order to induce an unnatural spawn. This was achieved by overall having a higher average temperature and on average being exposed to more light. So let's look at more of the numbers of what they were exposed to. The natural fish were exposed to 10.5 to 14 hours of daylight a day, whereas the artificial, during the months of March to September, received 12.5 to 14 hours of daylight, and from September to February, received 13 to 14 hours of a combination of natural and artificial light. Both groups experienced around 14 to 23 degrees Celsius water temperatures. The biggest difference is when they experienced 23 degrees Celsius. Natural, we hit that point in about late August and early September, whereas artificial reached it in February. Now let's look at how the GCMS is able to analyze the fatty acid concentrations. We took 248 samples, or 83 spawns divided into triplicates, and used a liquid-liquid separation. Like Nick said, we're going to go over how it's done. First, you take your sample and you'll add a 2% sulfuric acid solution in a solution that is a 3:2 ratio of toluene and methanol. This will separate the hydrophilic heads from the hydrophobic tails of the fatty acids. The samples are then added to a 100 degrees Celsius bath in order to accelerate this reaction. The samples are then removed. Hexane is added, which will join with the tails, and deionized water, which will join with the heads. This will cause the two layers to separate and have a fatty acid-rich layer on top and all the other parts in the bottom. We will then set it in the centrifuge, rotate it at 2,000 rotations per minute for 2 minutes, hoping it didn't break in the centrifuge as well. And then you will remove it, and you will have two very distinct layers. We will then remove the top fatty acid-rich layer and insert it into a GC vial. We then evaporate that sample, resuspend it in 1.5 milliliters of hexane. From there, we will take 500 microliters of that sample, add it to a new GC vial. Then we add 15 microliters of a C19 standard, evaporate it once more, and then finally resuspend it in 500 microliters of hexane. This whole process takes about a day to complete, and only then can you analyze it in the GCMS. The ramen, however, is a little more simple. You take your sample, you insert the probe, and then you acquire your data, which can take about 10 seconds. So let's see what the GCMS was able to find. This is a PCA graph. It's a multidimensional graph that allows you to compare many different variables at once. It can be seen that the natural samples are a little more varied, meaning that from sample to sample, they're a little different. The artificial is the opposite. They're a little more condensed, meaning that each sample is more similar to each other. You can also see there's only a slight overlap between the two groups, meaning overall these spawns are rather different from each other. So let's look at the numbers. This can be a little overwhelming, so I tried to color code it to make it a little more palatable. You can see that 15 of the 17 fatty acids that showed up in the eggs had a significant difference. C16-1 and C24 were the only two that had no significant difference. Also, when comparing artificial to natural, artificial spawn only surpassed the natural spawn in DHA, C24-1, polyunsaturated fatty acids, and omega-3s. So in conclusion, there is a significant difference in fatty acid concentration between naturally and artificially spawned eggs. So why were there differences? The differences could potentially be explained by how the marine fish utilize fat. In autumn months, it's been found that they typically have a higher fat concentration as they prepare to store it for energy and use it as insulation for the colder months and not really use it for reproduction. It could also be explained by the diet. One of the components, sardines, also have similar fatty acid trends, meaning in the winter months they typically have higher DHA and polyunsaturated fatty acids, whereas in the summer they're typically higher in saturated fatty acids. And how does this affect the egg quality? First, the artificial eggs were found to have a 75.1% hatch rate, whereas the natural had a 48.8%. And survival of first feed also differed, with artificial having a 61.8% and natural having a 74.1%. All other metrics, such as egg diameter, yolk diameter, were all pretty similar and showed no differences. And this is what the ramen was able to find. This graph shows how well the ramen was able to predict fatty acid concentrations. The x-axis shows what the GCMS measured, and the y-axis is the Raman predicted values. If we take a look at the R-squared, we can see that it reached a 0.922, meaning that the model has become very accurate. I was able to help contribute some data points to this graph. I was able to do the green squares, the blue triangles, and the yellow circles. Something that's interesting to point out is that just like the GCMS, it was able to distinguish the natural from the artificial spawn. So in conclusion, the rapid on-farm fatty acid predictive model using Raman technology did make improvements. So by adding more samples and increasing the integration time, the Raman model continues to become more and more accurate. So far, it is shown to be best suited for analyzing EPA and DHA along with polyunsaturated fatty acids. It has also demonstrated that just like the GCMS, it is able to distinguish different samples. So what's the big takeaway? Artificial spawning results in higher hatch rates and lower survival to first feed rates. So depending on the preferences of the farmer or what metrics they're chasing, artificial spawning can be a viable option for California yellowtail aquaculture. Raman has also demonstrated that despite being smaller, cheaper, and quicker, it is more than capable of being a suitable replacement for GCMS in aquaculture. I would like to thank the Link Foundation USD ARS for their funding and allowing students like me to participate in experiences such as this one. Thanks to them, I've learned how to operate many different instruments, dissect fish, and much more. I would also like to thank Kevin Stewart for the Hub SeaWorld Research Institute for the donation of the eggs used during this research, and thank Dr. Priscilla Winder and Dr. Tara Pitts for supplying us with liquid nitrogen. I'd also like to say thank you to Fernando, Allison, Ben, Paul, and the rest of Harbor Branch. Thank you guys for letting me join you this summer. I've been able to learn so much about aquaculture while meeting some of the kindest, most supportive, and brilliant coworkers that I'll probably ever have. And lastly, thank you to my new intern buddies. Being away from home for so long has definitely been difficult. Being surrounded by people who I can hopefully now call my friends made it a little bit easier. So that's all I have. Any questions?
Nice presentation. I just want to clarify, so the eggs that were donated by Hub SeaWorld, they use for the artificial and natural treatments. So they're from the same broodstock?
Yes.
Okay. And then another question is, what did you use to visualize your PCA? That was really, really cool, the little animation.
Allison was able to provide that for me. Yeah, so I would ask her about that. Anyone else?
Any other question for Jake? Go ahead.
Great presentation, great study, Jake. Do you think you might pursue the field of aquaculture?
Going into it, I really had no intention, but this has definitely opened my eyes to how interesting it is, so it's definitely a possibility. I'm still not really sure, but maybe.
Somebody else?
Great presentation. Can you tell me a little bit more about how the probe works? I found that really, really cool. Like, did you have, how it works?
So, I'm not sure if it makes a difference, but the way we did it in a dark room, and then you insert the probe into the sample, and the way it will shoot a laser, and the way that the light refracts off of the sample and how it analyzes that, it can determine what is like the contents of the sample.
Yeah, awesome. Thank you.
Yeah.
Somebody else? What is it?
Based on your experience with this, do you think that this technology is able to use it for the next species with really low levels of
I'm not exactly sure, but I would assume it would work the same way. So it may not be like as strong, but I'm sure it could pick it up.
Regarding that line of question, what other application do you see for this Raman technology in the agriculture world, for instance?
Not exactly sure. I guess just having a better idea for like farmers, whether or not to continue with the spawn that they have, if it's worth their time and investment to continue raising it and hatching it.
They can use it like to measure the fillet.
Oh yeah, I forgot. Yeah, you can stick it in the fillet as well.
Thank you. Okay. So our next speaker is Madeleine Tibes, and I read. Welcome in. Welcoming to the stage is Madeleine Madi Debes, who is a senior undergraduate student at the University of Maine studying ecology and environmental science with a concentration in marine ecology. Originally from Michigan, she has embraced her passion in exploring global ecosystem and working closely with the Fisheries, Ecology and Conservation Lab here at Harbor Branch. Welcome.
All right, welcome everyone. Since I've already been introduced, let's just get straight into this. So, what is a shell-crushing predator? Basically, the scientific term is durophagy or durophagous predator, which basically means that they can eat other animals that have hard shells or exoskeletons. And there's a wide variety of different durophagous predators across the animal kingdom. But specifically for my project, I just looked at these four, which is the Florida stone crab, the porcupine fish, the spotted eagle ray, and the loggerhead sea turtle. And each one of these have their own unique morphological appendages or just ways of eating hard-shelled animals. So for the importance, it is really important to understand how these animals interact in the food web, is specifically, so scientists can't follow each one of these animals or track them out in the field. So we have to use the power of technology and innovation to understand exactly how these animals are interacting with their prey, such as the hard-shell clam, which is what I used as a control in my study. And this can just help paint a clear picture of what's going on in these local habitats. It's also important because it can help current research, especially with the FEC Lab's research in identifying durophagous predators. We can understand the ecology of these predator-prey interactions much more closely by deploying hydrophones out into the field to gather more details on when and where these animals are feeding and why acoustics can finally give us a chance to really hone in on the predation process that's happening out in the wild. And it can also further build upon research to conduct predator monitoring programs that benefit research, shellfish farm operators, and restoration managers. So this is just a really great way to really observe these animals out in their natural habitat. So what is bioacoustics and what is a hydrophone? Let me explain. So bioacoustics is a way of studying how these animals sound or the sounds that these animals make. So I specifically looked at the feeding behaviors, which is what I focused on. And a hydrophone is basically an underwater microphone. And even if you don't recognize these concepts, I guarantee you've heard of them before, such as how do you even go to sleep at night when you don't have your whale sounds? Very restful. Or the thousands of hundreds of sounds of bird calls. So for my research objectives, I looked at two different objectives, which is looking at how the different predator species can affect the sounds of the clam. So can I tell what's eating what based off of the sounds of the feeding? And my second one is how the different environments affect the acoustic recordings. So can I tell the difference between an animal eating in captivity versus out in the wild? And so for my hypotheses, I said for my first part that there's going to be no significant difference between the four different animals. And the second part is that there's going to be no significant difference between the captivity trials and the field experiments. So as I mentioned, I focused on observing the foraging behaviors of these animals. So to do this, we placed clams in front of the individuals and they would crack down on the clams and the sound would be captured by our hydrophone recorder, which was within the area of the feeding. For my captive trials, I processed acoustic files that were previously collected by the FEC Lab at various facilities within the state, and each tank had an AS1 hydrophone and a GoPro Hero 4 camera so that we can confirm both the feeding on the hydrophone as well as the camera. For the field experiments, I had to kind of create my own setup for these sensors, so this is kind of how the one-eyed unicorns were created. These are sensors that are comprised of a cinder block base and a Snaps hydrophone and a Brina camera strapped there on zip ties. And for my first deployment, I had 3 different setups in the area of the Fort Pierce Inlet. And at each set, I put a, oopsies, I put a tray full of 50 clams about a 1 meter away. However, after looking at the footage as well as the recovery process, there did not seem to be any predation visible. So I kind of changed things up for my second deployment by adding 2 more setups, as well as went up to the Sebastian Inlet, so changed where I was located. And I also just laid the clams flat out on the sand, and we pre-cracked clams with markings on there, as you can see, and try to get the scent in the air, as well as we already saw some activity before we even left any of the sites, so that was really interesting. For my acoustic analysis, I used Raven Light for my analysis of the different characteristics of the acoustics, and I used Audacity to look at the different energy features of these events. So this is my comparison of the different events. So I looked at both the fracture or feeding events that happened. So this is like each clam that was being eaten. And then I looked at the shots, which are groupings of pulses or the fractures within the shells. So that was really interesting to look at. And so for my frequency analysis, I looked at these different... One second, things were going wrong. So I took the first shot of each feeding event and I put it into this graph so I can take a look at the different frequencies. So I took the peak frequency and the peak magnitude. So rolling into my results, my captive trials represented about 81 different feeding events. So these are from all the different species in total. So let's kind of take a look at what each animal sounds like, starting with the stone crab. So you can kind of see these shots are very far apart and they don't last very long. And the loggerhead sea turtle, very crunchy. And these shots are they last a lot longer. So the porcupine fish, however, they're very close together, which is a little bit of a difference. And the spotted eagle ray. Oh, what? We'll move on. Technology's not working today. So here is a representation of all the variables together, but this is kind of hard to look at, so let's kind of straighten this out or flatten it out. So this is a principal component analysis that's of all my combined data, and I had to kind of transform the variables so that you can have a better understanding. So these axes are driven by the different variables. So as seen on the x-axis here with a PC1, that's mostly driven by the number of shots, the event time, and the shot duration, which basically you can see that the stone crabs are kind of leading with that. It's pulling it more to the right, so that means they have higher variables, whereas everything else is towards the left because they have smaller variables in that in those durations. While the x-axis or y-axis has P2, and that's mostly driven by the shot rate, which is the porcupine fish, and the loggerheads have a lower rate. That's why they're kind of at the bottom, and the spotted eagle ray is kind of in the middle. However, if we change the dominant variables here, you can see that the spotted eagle rays actually have a high magnitude, while the loggerheads also leave with peak frequency. And heading back to my field experiment, the second one, I had to finally look at the cameras, and as you can see, there was a lot of activity going on, especially in the blue crab department. They were eating a lot of shells right in front of the camera, although I wasn't looking for them specifically, so kind of like they were mocking me, but anyways. But what about my 4 species that I was specifically looking for? I actually did find a confirmed spotted eagle ray feeding that happened on my 3rd camera, so let's try to see if this audio will work. very cool, very crunchy. It's what we like to see. And on my sites 4 and 5, I actually had 2 feeding events that happened at night, so the cameras were already turned off. So I quickly compared this to a captive trial of a spotted eagle ray. So we think these are spotted eagle rays based off of just the event times as well as the peak frequency. The peak frequency was within that 1 to 10 kilohertz range, so these are very similar. Heading into my discussion, since the Florida crab did have the longest event times in the shots per event, and they had the longest pauses between each shots. So, and the loggerheads had the longer shots and the highest peak magnitude. The porcupine fish had the highest shot rate, and the spotted eagle ray had a higher peak magnitude, which is leading me to reject the null that there was a significant difference between these different species because there was a significant difference between these specific sound characteristics I tested for. So I can positively say that you can identify these species based off of these specific acoustic features. And my second part, I think you can definitely accept the null on this between the 2 feeding events that happened at night and the 1 feeding event that was a captive one. They did have a significant similarity, so I would like to tentatively accept this null because of those 2 blind feeding events. And lastly, for future research focus, I would suggest, make all these suggestions. And lastly, all the data that was collected through this project will be used in the creation of an AI-generated detection classification program to identify these Dirophagus predators based on these manually produced key characteristics. I'd like to acknowledge everyone that was involved in making this process, this whole project possible through data collection, funding, resource providers, as well as in the field hands-on help. And that's a crack. Thank you, everyone. And are there any questions? Yeah. So it's because of the amount, the cameras were programmed to shut off after a certain amount of lumens were detected. So if there wasn't enough light, the cameras shut off. It was kind of like a battery and memory storage sort of situation, but it just made the cameras last a bit longer in the field. But yeah, you can't, even if the cameras were on, you wouldn't have been able to see anything because it's just going to be pitch black. Thank you. You can kind of tell what this noise was. We kind of chose areas that were not high in boater activity, so we didn't have a lot of boating activity, so you couldn't hear the motors. If there was anything that was too strenuous, I just manually went through and checked to see what that was, and I would check the cameras. So it's kind of one of those situations, just checking each one against each other.
Mike? Yeah, I was curious with your one on-camera feeding event with the spotted eagle rays, it seemed like a lot more crunching than other recordings I've seen.
Yeah, so that was actually the problem with that. That camera, that event, most of the captive trials, we put 1 clam into the tank and they would eat that 1 clam. This guy took 5 clams at once and decided to go ham on it. So that's the difference. That's why I couldn't compare that to the captive trials, because there was more clams, and so that's a different variable that you'd have to look into. Yeah. Yeah, so we actually have people in the lab working with the aquaculture farmer that's up in the Sebastian, and they've actually seen predation events on their clam leases. So some of their gear has actually been damaged because some of these predators have been eating or foraging. So this is another way to use hydrophones to try to track like what's going on. Are these animals feeding on those clam leases? So this is just another opportunity to use technology to observe these guys out in the field. So yeah.
Any other questions?
Thank you.
So we are going to finish our session with the last speaker, Emory Tudor. And Emery is a biology and English double major going into her senior year in Amherst College. She is obsessed with wildlife and spends most of her time at Harbor Ranch out on the boat, sorting through algae, in the lab experimenting with sea slug, and too close to the campus ponds, searching for gators. Did you find any?
I did. I found 3.
Hi, everyone. So my name's Emery. I worked this summer with Dr. Rachel Bruton to study the herbivory of Elysia subornata on Caulerpa prolifera in the Indian River Lagoon. Basically, I call it the Great Slug Search. I want to start with a story, a slug story. In the late 1980s, there were vast Caulerpa meadows in the IRL. These are quite similar to how they are today, where in areas that seagrass had declined or where there was bare sediment, this native green macroalgae had come in and colonized these areas. It looks much like this image on the left. These meadows provide critical ecosystem services, including supporting fisheries, nutrient uptake, and sediment stabilization. And while they're not as good as seagrass per se, they're better than, a bare bottom. In the late 1980s, specifically winter of 1986, these meadows began to mysteriously disappear. Scientists would go out to these meadows, which had previously been, quite lush, and they would find root nubs and a bunch of dead slugs. They figured out pretty quickly that the culprit was Elysia subornata, a tiny green succoglossin, aka a sap-sucking sea slug. It's like a little rhyme in your mouth. And they're about this big, and they are voracious. They ate about 70% of the Caulerpa prolifera in the Indian River Lagoon in just 7 months. And then about a year later, they entirely disappeared, like almost no trace for about 40 years, actually. Scientists speculated that it might have been some sort of, water quality decline or maybe a population collapse, but they really didn't know, and there weren't extensive studies, and because the slug remained essentially absent from the IRL for 4 decades, they really didn't have much to say on it. However, they're back. They started showing up recently, and so that is what prompted my project. A little bit first on why we actually care about these slugs, aside from, the fact that they eat all of our Caulerpa. First, they are really interesting specialists. You can see from their camouflage that they're quite adept at blending with Caulerpa prolifera. That's 2 photos I took using our ROV on the bottom right, and you can tell that they look almost exactly like the leaves they feed on. They're also super interesting because they're kleptoplastic, which means that they steal chloroplasts from the algae they eat, hide them in their bodies, and then essentially, use sunlight to make energy. They're basically like a plant. They look like one, act like one. And you can actually see from the images on the top right, which I took using a dissection microscope, that the concentration of chloroplasts changes their color. So the dark green one on the top, that one has more chloroplasts. They're the tiny little branching molecules, while the one on the bottom left is lighter yellow in color and has far fewer chloroplasts. That one was starved for a few days, so it started using those chloroplasts up because it was trying to get energy through an alternative method. So we were looking at a site in Palm Shores that we believed the slugs had just arrived to because we found them in quite low densities compared with other sites that had, just the dead slugs left. We hypothesized that as the slugs colonized the site, slugs' density would increase across all depth zones and the percent cover of the Caulerpa prolifera would decrease. In order to test this, we had 2 different types of methods. We had field work and lab work. In the field, we would go out every week and we would use a hanometer to take water quality data. We would do dip netting for 10 minutes where we'd just sort of lug around these massive nets in the water and catch as many slugs as possible. And then we would do ROV surveys to monitor the Caulerpa cover on the bottom. And then in the lab, we would use slugs that we collected to conduct feeding assays. These helped us determine grazing behavior and rates. And then we would do imaging as well, which helped us get, those images that you saw before of the slug flesh and the chloroplasts inside. Our site was about an hour-and-a-half drive north of here in Palm Shores, right near the bridge. And we had 3 different depth zones that we were doing sampling at: a nearshore, a midshore, and an offshore. The nearshore, which had the lowest Caulerpa coverage, about 50% at our beginning, was about 1.5 feet deep. The midshore was about 2 feet deep, and the offshore was about 3. And then midshore and offshore both had 100% cover, like super great beds, and more slugs than the other site. So we expected that, slugs would arrive to the offshore and sort of colonize inwards as they went. So this is us being slug wranglers. We would go out and we'd dip net, we'd dump it all into that blue bucket there and sort through by hand. If you've ever just sort of stuck your hands in a dark, muddy bucket and felt something squirming around, it's really not the most pleasant, but it's actually quite fun. You could tell that the slugs were there because you often couldn't see them very well except for a little bit of a blue sheen from their mucus, but you would feel them. They're very slimy, so you could tell they're there. Just you'd reach your hand in the mystery bucket and you'd figure it out pretty quick. So we often would collect them and we'd get buckets like that and we'd do counts at each site. And then we would actually steal them. We'd take them back to our lab. This is me with my slugatorium. We had about 100 to 150 slugs at any given time in there, and that's where we would experiment on them. Not in a bad way, it was very humane. And you can see that photo on the right, just how similar they look to those leaves. That's a mix of slug and leaf. I think in the photo there's about 13 slugs in this photo, but you probably won't be able to see them all. They look exactly like the leaf, it's ridiculous. In the lab, we did 3 different types of experiments. Starvation trials with 0, 24, and 48-hour treatments. These were designed to test whether or not grazing rate was impacted by starvation. If they're more hungry, are they going to eat faster? Then we did mixed algae trials where we would take a Caulerpa prolifera leaf and a little bit of the drift algae, Gracilaria tekvahi, and we would test whether or not it would eat the drift algae as well as the leaf. And that was because we would often see in the sites that the slugs were on these giant clumps of drift that would sort of move around with the currents. And we expected that might have been a way that the slugs were moving between different sites, or, if they were sitting on it, maybe they were eating it. We didn't expect that, but we wanted to test anyway to ensure. And then we also did preference trials where both those types of algae would be in the same container. We found that starvation was insignificant at this smaller time scale. We think that because they can live up to 25 days without food, it probably just wasn't long enough. And then we found also that Caulerpa prolifera is always preferred. They did not eat the drift at all. Maybe if, again, they're 25 days starved, they would munch on it a little bit, but they really didn't want to touch it at all. And the way we looked at feeding also was you could see these really obvious bite marks. Because they're sacoglossans or sap suckers, they would bite onto the algae and take just a full chunk out and leave that really distinct marking. Using these markings, we were able to get a general grazing rate from our experiments. We did 75 trials total and found average area consumed over time. We used the number of bite marks on the leaf and then the percent area of the leaf consumed. This was all measured in image J. And we got an average rate of 1.2 centimeters per hour, centimeters squared per hour. This is much higher than previous reported rates of 5 to 6 centimeters per day. Ours is about 29 centimeters per day in comparison. But we believe this difference comes out of the fact that the Thibault M&S trials were doing group trials where they would take, just a giant handful of algae, dump it in a bucket, and then throw a bunch of slugs in with it and just leave it until the slugs finished eating the algae. Whereas ours was a single leaf, a single slug, and monitoring how much it was able to consume in a 3-hour time period. So while theirs might be more ecologically relevant, ours is a bit more relevant on the individual scale. Everything was going great with our project until the plague. You can see here 3 of our poor slugs have succumbed. They basically just dissolved. We really weren't sure what was happening at first. We brought back a fresh batch of slugs and put them in our tanks, and we came back the next day and half of them had died. And then the day after that, they were all dead. So very rapid, essentially extermination of our slugs. We quickly cleaned out all of our tanks, did a full reset to make sure it wasn't something we were doing with the filters. We didn't expect it to because we already had slugs living in those tanks doing totally fine. And we got a new batch of slugs the next week, and they all died as well. So at that point, we stopped bringing slugs in and stopped doing our feeding trials because they just weren't surviving, and I felt kind of guilty. I was a bit emotionally attached to them. They're so cute. And so I felt bad having them end up like this. It also smelled really bad. So the plague really put a damper in our field trials, and we definitely expect it to come that it was a wild pathogen. That is in part supported by our field findings. As a reminder, we expected all of the populations across the different depth zones to continually increase. There was plenty of chlorpa for them to feed on. We saw evidence of lots of eggs, lots of juveniles. There shouldn't have been an obvious reason for their populations not to continue increasing. But you can see at about week 5, we had massive declines, especially in our deep site, which had the best chlorpa of it all. So we expect that there was some sort of pathogen that was dampening their populations in the wild, especially those adult populations. Interestingly, we did have a juvenile spike, especially in the midshore site on our final week. You can look on that graph on the right. So it is possible that we'll continue to see increasing populations or recovery going forward. It's just unclear, these slugs are quite, novel in their appearance, so any pathogen affecting them is really something that's never been encountered or recorded. So we did not see the change in chlorpa cover that we expected due to Elysia subornata herbivory. However, that's probably due to a number of possibilities. One, it could have been too short of a time scale. We were only looking at about 7 weeks, so it's possible they just didn't have time to get up to that high population density to eat away all the chlorpa. In the 80s, they were reporting 100 per square meter. We did not see anywhere near as many as that. But then it's also potentially the plague that dampened down their populations enough that we weren't able to see that continued population growth and herbivory. We did have some natural senescence, so the nearshore site, which was the shallowest one, declined from about 40 to 50% cover down to 0. But that was just, there were never a huge number of slugs discovered here, so that's just the natural death of that algae. But the offshore and the midshore sites remained at this about 100% cover. You can also see a bit of the, I forgot about this, but my advisor discovered the other day that there is a slug in this photo. That tiny blip. I didn't even notice until she pointed it out. They're really hard to see. We think that there's still potential for collapse in the Palm Shores site chlorpa beds, but it's unclear whether or not that will be, soon or farther in the future. Especially if this pathogen continues to affect their populations there, it could be a much longer time scale. Future directions for this should definitely include continuing monitoring across IRL sites. One of the sort of drawbacks in the 80s is that they only had monitoring at patchy sites, and it wasn't very reactionary to the increasing slug populations. We have the chance now to work with other institutions to get monitoring across the IRL so we can better understand the population dynamics and how the chlorpa beds are being affected, especially because if these chlorpa beds disappear, and especially with our low levels of seagrass right now, it could really impact things like blue crab, snook, other fisheries that people in this community really rely on. And then we should also establish aquaria for the slugs. If they disappear like they did 40 years ago, we're going to have sort of no way to continue studying them. Having them in captivity is going to be really important. And we've actually already started planting chlorpa in our seagrass nursery so that we can have ways to feed them in the future if we can, get some slugs without disease to actually stay alive again. I want to thank everyone in my lab for both showing up today. I really appreciate you, and helping so much with sorting through algae or helping me in the lab. I appreciate it so much. And then to the Link Foundation and the Harbor Branch Institute Foundation for funding this research. I had a great time going out on the boat and collecting slugs. I would not have expected that ever, but I loved it. So thank you to everyone. And does anyone have any questions? Okay, so 40 years ago, they speculated that it was due to declining water quality because other slug species declined around a similar time. I don't really think that's accurate. These slugs are really hardy. I accidentally left one, clinging onto some chlorpa in a bucket, at 17 degrees for 2 weeks, no aeration. It was fine, fully fine. So I think that was not the cause. I think it's probably they had such high population, ate all their food, that they just had a massive crash, and there were only a few left over and not enough to really bring it back, especially because it took about a decade for the chlorpa to recover. So they just didn't have that high food quality.
Nice talk, Emery. I'm curious about your decision to use ROVs at such shallow depths. Can you talk us through why you chose that versus just like snorkeling with a camera?
Yeah, okay.
And number two, do you have any idea whether or not this species might be simulparous? Might it just reproduce once and die?
In terms of, let me answer the first question. In terms of ROVing, we did that because the water, especially at the beginning, was really low quality. Like it would give you an infection if you were getting in there for a while. It was quite gross. And there was even a sewage spill like a couple miles down the way. So we were like, nah, it's fine to leave it in the water like that. So we did the ROVing. And then in terms of simulparity, they don't seem to be simulparous. When I had them in captivity, they were continually producing eggs. And they live for about 240 days. So they have a pretty long life cycle for a slug. And they can reproduce anytime after 75 days. I'm 99% sure they reproduce multiple times. And with lots of eggs in each. They lay these like coils of eggs. Wait, actually, I think I have a photo. Boom, 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 boom. Okay, yeah, this like yellow coil here, it usually has like 300 eggs, but it can be up to like 1,200. So yeah, they reproduce quite a lot. It was actually really annoying. They were laying them all over the side of the tank. I would like have to like scrub them off. But I was like, sorry, baby slugs, like you don't get to make it.
Another question? Do you know how they, or if they sequester the chlorophene, or how they deal with them at the time?
Yeah, they just keep them in their body. They're super toxic. They don't really have any natural predators because of that. We didn't see any predation as well. Like when we would dump the slugs back in the water, they would just float to the bottom. Nothing wanted to eat them. We actually did see an example of when we planted the chlorpa at the seagrass nursery, one slug like made it in. And it didn't make it very far, actually. One of the sea hares ate it by accident, and that sea hare just died immediately. Like, I actually found the dead sea hare with the dead slug inside of it. It was very, very gross, actually. It smelled terrible. But they're super toxic because they just sequester those chlorpenines in their body.
My question was about the predators. But
Yeah, I think also it's possible they have some predators, but I just don't think that predator would do very well. I think they would die.
So they have a pathogen, or that's what you think there's a pathogen.
Yeah.
So are you looking at the genetics of the whole thing to see what that pathogen might be?
We did not have the chance to look at the genetics. I think there's still the possibility of like if we go out again or if this research continues to look at that and do the genetic part. But we were just mainly focusing on the ecology. So it's definitely an avenue that warrants exploration, though.
Amanda?
They'll eat any species of chlorpa, and most of the previous research on them has actually focused on using them to get rid of chlorpa taxifolia in the Mediterranean, but they don't like the cold water, so it doesn't work out so well. But yeah, they can eat any of the species. It's just that Chlorpa prolifera is the one that forms, those robust beds compared with the other ones. Oh, I bet you they would if they encountered it, but because it's not quite as abundant as these beds, they seem to mainly be in the beds here where it's just like easy to like float around, and it's the chill life as a slug, I think, until they eat it all and they starve to death. But until then, it's like good life. Yeah.
Any last question? Well, good job. Now I think we'll have a break and please join me to do the rest.
That was great.
Oh, yeah, I see.
Wait, is it on? He might have not on. I mean, he might not have it on. Yeah, this will change it.
Oh.
Now
Or
I
does the
can-
laser work?
It should. Okay. So now it's on. Oh. That's a full battery. So the laser works.
Okay, yeah. That's honestly all I need.
He might be
I think
busy
so.
with that.
Should I
Sorry.
put you in?
We're not getting started.
Wait, can I?
Yeah.
Oh, no.
Sorry. We should have called you.
Okay.
We're raising them. Yeah, it's
Wait.
on and it's working.
Okay, yeah, we're doing it.
Okay. So I don't know what Amy wanted. So we're going to continue with the afternoon session. And thank you all for coming and joining us. I think so far the presentations have been amazing, and I hope this afternoon it was going to continue to be the same. Our first speaker for the afternoon session is Ms. Christina Xu. She comes from the University of New Mexico, and she's going to be presenting on the prevention of pancreatic cancer using marine natural products. And I was her mentor.
Hello. So like Esther said, my name is Christina Xu, and this summer I was in Dr. Guzman's lab, and my project was on the prevention of pancreatic cancer using marine natural products. So cancer is actually the second leading cause of death worldwide, and in 2020 alone, it cost the U.S. $208.9 billion. Now, this comes from things like healthcare costs, medical costs, treatment costs, and even lost earnings. Pancreatic cancer specifically is one of the most lethal. It's the third leading cause of overall cancer deaths, and only about 13% of patients survive 5 years after diagnosis. And one of the most important risk factors for pancreatic cancer is pancreatitis, or inflammation of the pancreas. And a lot of new studies have shown a correlation between mast cells and degranulation with the onset of pancreatic cancer. So what exactly are mast cells? They're immune cells that develop to have these granules, which are basically just pockets of biologically active molecules. This can be things like signaling molecules or growth factors. And so when your mast cells are signaled to, they will release these products in a process called degranulation. Now, this results in a plethora of different responses, including tissue repair, mediation of inflammation, pathogen defense, and even just your everyday allergy symptoms. So to kind of tie everything together, the inflammation caused by pancreatitis recruits mast cell migration to the pancreas. And not just this, but it promotes them to degranulate. So this sort of accumulation of degranulation products changes the microenvironment around the cells to be one that's conducive towards the onset of pancreatic cancer. And a lot of studies have also shown that these products are tumorigenic, meaning that they actually promote the growth and metastasis of existing cancer as well.
So obviously that's not a good thing, and there are two sort of logical ways to prevent this. One is to stop the mast cells from migrating to the pancreas in the first place, and the second is to stop the mast cells from degranulating. And my project specifically focuses on this, so the inhibition of mast cell degranulation. And we hope to do this through the use of marine natural products. So these are compounds that come from marine organisms like sponges, corals, algae, and bacteria. And they're really cool because, number one, they're diverse because the ocean's environment is so diverse in things like pH, sunlight levels, and temperature. And something that's also really cool about them is that they're all biologically active. So we know this because, number one, they come from non-motile organisms, meaning they don't move, and their only form of communication, competition between each other, and defense against predators are these chemical signals. Two, they are secondary metabolites, meaning they are not required for survival, but they still evolve to have a selective advantage for these organisms. And three, our sponges and corals are exerting energy to make these products. So it's kind of counterintuitive if they're wasting all this energy making these products when they don't even do anything. So all of these things that I just mentioned make them really good candidates for drug discovery. So our hypothesis is that we can use marine natural compounds to inhibit mast cell degranulation. So Dee from my lab did a lot of the groundwork for the assay that I was going to be doing this summer. She implemented it from rat cells into a human cell line, and it was my job this summer to go from a 96 well plate into a 384 well plate. Now this is really important because this allows our screening to be a lot more effective because we can screen more compounds per plate, making it take less time. And while this seems kind of trivial, it took actually a lot of troubleshooting, and something that kind of added to that complexity was I had to use this machine, the Integra ViaFlow, and it's basically an automated pipetter. So you program it, and it basically pipettes everything in your assay for you. And the really cool thing about this is that it takes out the human inaccuracy of pipetting, so it makes our assay more accurate, which is really good. So I've been kind of talking up our assay, and I know this figure is kind of daunting, but I'm going to kind of walk you through what I did. So when we test our marine natural compounds, we actually test them as enriched fractions, which is just like, it's just multiple compounds within one sample. And so our first step is to plate our human mast cells or LUVA cells. And this just means we're putting cells in each of the wells. And then after that, we actually do this to 2 plates because we run 2 assays, one for degranulation and one for cytotoxicity. So I'm first just going to talk about the degranulation, so this top part here. And so the first step after plating is to add our marine natural compounds, and then to half the plate we add buffer so we can measure basal levels of degranulation, and then to the other half of the plate we actually add calcimicin, which induces mast cell degranulation so that further along in the assay we can determine levels of inhibition. And then after that we add PNAG and carbonate. And to kind of explain these 2 steps a little further, our assay is a colorimetric assay, meaning that we expect a color change as a result, and we're determining levels of beta-hexosaminidase, which is an enzyme that gets released during degranulation. So we add PNAG, which is the substrate for our target enzyme, and what happens is if any degranulation occurred, our enzyme will just be floating around in the well, and it will bind to PNAG, its substrate, and create this product, 4-nitrophenol. Now, at this point, we add carbonate, which has a very basic buffer. So number 1, it stops the reaction because it takes our enzyme out of its optimal pH range. And number 2, it deprotonates and ionizes the product. And this is actually what causes that color change, and it turns yellow. So in this case, if you have a more yellow color, that means more degranulation. And if you have a less yellow or more clear color, that means there was less degranulation. And this is kind of what your plates end up looking like. I know it's really hard to see, so you're going to have to kind of believe me here. But on the left, I have boxed wells that are more yellow, meaning more degranulation. And on the right, I have wells boxed that are less yellow, meaning less degranulation. And so while our eyes can't really see the difference between this, we put these plates through a plate reader, which is really sensitive to these like small color changes. And to kind of explain our cytotoxicity assay, this measures levels of cell death.
And this is really important, especially if we want to use these compounds in a clinical setting, because like I said before, our mast cells are really important to our innate immune system, so we don't want to just completely get rid of them. So after we plate our compounds, or plate our cells, we add our compounds, we add media and MTS. So to kind of explain that a little further, MTS is actually yellow in color, and so after we add that to our cells, if there are any that are live and metabolically active, they have enzymes that will turn MTS into formazine, and formazine has a sort of purple color. So if you have a more purple color, that means you have more active cells and less cytotoxicity, or less cell death. And in the other case, if it's more yellow, that means there are less active cells and more cytotoxicity, or more cell death. So this is what your plates end up looking like, a lot easier to distinguish. So on the left here, I have wells highlighted that are more purple, meaning there are more live cells, less cytotoxicity, less cell death. And then on the right here, I have wells that present as more yellow or orange, meaning there are less live cells, more cytotoxicity, more cell death. So like I said before, after we get these plates, we put them into a plate reader, which basically spits out a bunch of absorbance numbers, and we input that into Excel, we mess with some of the numbers, and we get these graphs. And I know it seems really overwhelming because we have so many samples in one plate, so I kind of cut these graphs in half so that we can actually look at what's going on. So here, our first graph, on our x-axis, we have our fractions labeled. And to kind of walk you through the controls, NT stands for non-treated, or nothing extra was added to it. And then we have two solvent controls, ethanol and methanol. And those two are used because that's what our compounds are dissolved in. And then finally, I have a positive control, which is apigenin. And that's used because it actually has been shown to inhibit degranulation. So on the y-axis, we actually have percent inhibition of degranulation. So if you take a look at our very first compound, it has a very negative number. And what that means is that it actually promoted degranulation, while the two compounds next to it are above the zero, meaning that they promoted degranulation. And so for our purposes, we wanted to find compounds that were at least 50% inhibition of degranulation, and that's what this dotted line here represents. So anything above that dotted line would represent potential hits. And so for the cytotoxicity, it's kind of the same story, except on the y-axis, you have percent cytotoxicity, or percent cells that have died. And so for our purposes, we wanted less than 20% cytotoxicity, and so that's what this blue dotted line represents. So any compounds that are below that dotted line would be good candidates for potential fractions. And so in total, we tested 256 fractions, and we had 4 hits, and these two graphs here are just comparing those 4 hits. And actually, our third compound was the most efficient, with a 91% inhibition with nearly 0% cytotoxicity, which is really exciting. And so all of these were actually sampled from sponges. So these are the actual organisms that our fractions came from, which is really cool. And it's amazing that we actually found compounds that, inhibited degranulation, but there's still a lot of work to be done. So in the future, we hope to test more fractions, and with the fractions that are hits, we hope to purify them into single compounds so that we can do further testing and figure out what mechanisms they do to inhibit degranulation. And not only this, but we hope to implement a flow cytometry assay to be able to validate our findings. And way into the future, we hope to actually apply this assay in tumor cells so that we can determine the effects of our marine natural compounds on cancer progression and metastasis. So I would like to thank the Gertrude E. Skelly Charitable Foundation for funding my project, as well as a discretionary grant by the Harbor Branch Oceanographic Institute Foundation. And of course, a big thank you to everyone that was in my lab, Esther, Dee, and Tara, for being amazing mentors throughout the whole entire summer. And of course, the other interns for making the experience really fun. Any questions? Hi. Thank you.
So I have a technical question.
Yes.
Why you
before inducing the degranulation of the mast cells and not the other way around?
Well, actually, when we're performing the assay, we add them at the same time. So we add marine natural compounds, and then we add the calcimicin, and then we mix it, and then let it incubate. So it's not like we don't incubate it with our marine compounds and then add the inducer. They get added at the same time. Yeah.
Okay, perfect. Thank you.
Of course. Oh, the fractions, they actually come from the organic chemistry lab. So Jenny's going to tell you a little more about that, but they actually like purify and extract stuff from our organisms. So, oh, or do you mean like where geologically?
Yeah, where did the organisms come from? Do you know which, like the ones you tested, where they came from?
I know some of them came from a Puerto Rico cruise, and then some of them also came from Florida, but I'm not exactly sure where in Florida they came from.
Okay, perfect. Thank you.
Yeah.
Anybody else?
Have any of those 4 compounds had hits against anything else other than this particular pathway?
I'm not sure if we've tested those fractions in anything else, but I think a lot of marine natural compounds tend to have like multiple effects, so that definitely could be something to look into.
Any more questions? Okay.
So the fraction that induced granulation,
Yes.
seemed to induce it really well.
Yeah.
It's out of my field. Is there any reason why we wanted to induce granulation?
Yeah, so there are, since we're specifically looking at cancer, we want inhibition of degranulation, but since there are a lot of other diseases out there, there are some that require like promotion of degranulation to actually like prevent those things. So it's important to keep track of also the ones that promote degranulation as well.
I think you did a great job, so please help me.
Thank you.
Our next speaker is Jenny Shah from the Indian River State College. Her talk is going to be the discovery of marine natural products from an aplycine sponge with antibiotic effects. Her mentors were Dr. Priscilla Winder and Dr. Amy Wright.
Good afternoon. Like Esther said, I've been working with Dr. Priscilla Winder and Dr. Amy Wright in the Chemistry BMR lab. Their research focuses on isolating compounds produced from sponges with antibiotic effects. My project has been to test the 128 sponge samples collected from their 2024 Puerto Rican research cruise for their antibiotic activity and select one species to isolate pure compounds from. Sponges were selected for study as their compounds have shown to have antibiotic effects in prior studies. As sessile organisms, they need to produce compounds to defend themselves from threats on the reefs. And as filter feeders, they also host a variety of symbiotic relationships with bacteria and fungi, both of which contribute to their interesting chemical and biological properties for use in antibiotic development. We're specifically looking for compounds active against MRSA, methicillin-resistant Staphylococcus aureus, a type of bacteria resistant to many antibiotics. MRSA causes serious skin infections, which can worsen into pneumonia, blood infections, sepsis, and death if left untreated. This research is important as many bacteria develop resistance to antibiotics used in clinical practice for various reasons such as overuse or inappropriate use. Therefore, there's a need to identify new antibiotics to continually fight bacterial infection. Currently, over 70% of drugs and antibiotics are derived from terrestrial sources, and rising antibiotic resistance encourages the search for alternate sources, such as the ocean, which is home to many organisms that produce a variety of secondary metabolites with strong biological effects. All 128 extractions were tested in a distillation assay, a qualitative assessment of antibiotic effectiveness. Each sample was pipetted onto an individual paper disc and placed on agar seeded with MRSA and incubated for 24 hours at 37 degrees Celsius. After the incubation period, we measured the zone around the disc where the bacteria couldn't grow due to inhibition to that particular compound. The large zones can be interpreted as that compound diffuses well through the agar, it's bioactive, and the bacteria are sensitive to it. The largest zone produced was 27 millimeters by a new species in the Aplycina genus. This was the sponge I continued with throughout this project. The Aplycina sponge was collected off of the west coast of Puerto Rico in the Mona Passage. On the right is a photo of the sponge taken from the submersible, and on the left is a photo after it was taken up to the ship deck. Before isolating compounds, we needed to prepare an extraction of the sponge. A piece of the sponge was removed from the bunker, then blended with 300 milliliters of ethanol, and then allowed to steep overnight in the freezer to allow the compounds to draw out into the solvent. Ethanol is a polar solvent that helps to separate a wide range of compounds and helps to lyse the cell membranes to release those compounds. The next day, the extraction was run through a filter apparatus to separate the liquid extraction from the sponge tissue. After that, any remaining bits of solid sponge were further blended with ethanol and filtered again twice. Any remaining bits of sponge after that were then blended with 300 milliliters of ethyl acetate, then filtered twice. Ethanol helps to draw out the more polar compounds. Ethyl acetate helps to draw out the less polar compounds left behind. The extraction was then dried down under rotary evaporation to gently and efficiently remove solvents under reduced pressure before being transferred into a separatory flask for solvent partitioning, which allows us to separate a mixture into different parts based off of how different compounds have different solubilities and polarities in either of one or two immiscible or unmixable liquids. The extraction was partitioned 4 times with 100 milliliters of water and 150 milliliters of ethyl acetate. Then the water partition was further partitioned with butanol, leaving us with 3 partitions, ethyl acetate, butanol, and water with separation of compounds in each. To decide which partition to continue isolating compounds from, one technique we used is HPLC, high-performance liquid chromatography. Chromatography is a technique to separate compounds in a mixture by running the compound in a mobile phase through a stationary phase. In HPLC, the stationary phase is the mobile phase is pressurized and pumped through the system. The sample is then injected and it gets carried through the mobile phase into the column, the stationary phase, where separation of compounds occurs due to the compounds' different affinities to the column. Then the compounds get passed through the detector and display on our computer as a chromatogram. And how long the compound stays in the column is called its retention time, which is unique to individual compounds, allowing us to identify them. Prior to chromatography, we confirmed in another distaffusion assay that of the 3 partitions, only ethyl acetate was active, in addition to the ethanol crude, of course. And then after that, we looked at the chromatograms, comparing the butanol and the ethyl acetate chromatograms to the original crude extract. We see a significant peak around retention time 9, highlighted, that separated out nicely into the ethyl acetate partition. So the assay tells us to continue with the ethyl acetate partition, and the chromatograms demonstrate a significant peak of interest for pure compound separation. Then 1.5 grams of the ethyl acetate partition was run on a flash chromatography system called CombiFlash, which, like HPLC, uses high pressure, but it uses reduced pressure and is a fully automized system to collect separation of sample into a variety of test tubes. So the 93 test tubes collected were then combined into 16 fractions into 1-gram vials based off of how multiple samples in different test tubes can correspond to a peak, as shown in the highlights. Then all of the 16 fractions were tested in another distaffusion assay, and there was a variety of actives. We chose the largest zone of fraction 5 to continue with. Fraction 5 was then further fractionated or separated using the preparative HPLC on the water's liquid chromatography system, which is a form of chromatography that uses higher resolution than the CombiFlash by using a smaller column to separate smaller amounts of liquid with higher resolution. So we use CombiFlash for initial solvent partitioning, then waters for any further automated partitioning we need. On the left shows the peaks separated at different retention times. In the middle, we have graphs that tell us which tubes each peak separated out into, and then the fractions were like Combi flash combined, or the samples were combined into 10 vials based off of how multiple tubes correspond to an individual peak. The fractions were then tested again in a distaffusion assay with sample 7 showing the most antibiotic effect. So sample 7 was selected for compound identification using nuclear magnetic resonance, which is a technique to determine the structure of a molecule by applying a magnetic field to analyze the spin property of atomic nuclei, the center of an atom containing protons and neutrons. Each atom demonstrates its own unique chemical shift, the frequency at which the nuclei absorb the magnetic field and spin, called resonance. Chemical shifts are displayed as numbers on the bottom of our graphs. Proton and carbon NMR hint to how many protons and carbons there are in a sample. We estimate about 13 protons and 16 carbons initially. There could be multiple under an individual peak, which more advanced NMR will inform us. Proton NMR demonstrates peak splitting, so there could be singlets, doublets, triplets. There's 2 doublets in this sample, which means that one proton is recognizing 1 proton on a neighboring atom, so this information helps us to piece our structure together. One type of advanced NMR is called COSY, correlation spectroscopy, which shows which protons are coupled or located on adjacent atoms. There is a proton at the 6.15 chemical shift that recognizes a proton on the 3.88 chemical shift. Both axes show proton, and vice versa. The 3.88 on the X recognizes 6.15 on the Y. So this tells us that these 2 protons are adjacent, and in the previous slide, both of these were the doublets. And on the X equals Y axis, protons also recognize themselves. So HSQC is another form of NMR which tells us which protons are directly bonded to which carbons. So the x-axis is protons and the y is carbons. The 3.88 proton recognizes a carbon at 77.43 chemical shift, placing that proton on that carbon. And then HMBC tells us which protons are recognizing carbons 2 to 3 bonds away from that proton. The 6.15 proton recognizes the carbon at 77.43, which is 2 bonds away. And the 6.15 also recognizes a carbon at 113, which is 3 bonds away. Putting together, after fully analyzing these advanced types of NMR, it allowed us to put these partial structures into complete structures. We then realized that there are 2 pure compounds within the fraction 7 sample, which took a few weeks to identify as they were solved within the mixture. After we identified the compounds, they were then manually separated using HPLC, and further NMR confirmed that they separated out correctly. We ran these compounds through databases and found that they have already been identified. After they were separated, they were further tested in a broth solution to determine the minimum concentration required to inhibit the growth of bacteria. On the left, aeroplazinin 1 has an MIC of 6.2 micrograms per milliliter, which in comparison to one of the controls, chloromethanicol, which is a drug known to still work against MRSA, that MIC is 3.5, showing that aeroplazinin is actually quite effective. In comparison to the structure on the right, whose MIC is greater than 50 micrograms per milliliter, which is much less effective, comparable to methicillin, which MRSA is known to be resistant against. Now, these arrows demonstrate, yes, the HMBC correlations, which significantly helped us put these structures together by understanding which protons are signaling to which carbons 2 to 3 bonds away. So in conclusion, there was 128 species that were tested for their antibiotic activity. Many species showed to be antibiotically active. 1 species was selected for pure compound identification. 2 pure compounds were isolated and their structures identified. Next steps would be for the isolated compounds to undergo further testing for their potential for use in antibiotic development. And the Harper Branch Chemistry Department will continue testing the remaining bioactive sponges from the 2024 Puerto Rican cruise. Thank you to the Gertrude E. Skelly Charitable Foundation for funding my project and making my amazing summer experience possible. And thank you to my incredible mentors, Dr. Priscilla Winder and Dr. Amy Wright, for your guidance, your expertise, your patience in selecting me to be your intern. And thank you to Deidre Harmody for your continued support throughout my internship. Thank you all for your time. Are there any questions?
Super impressive work.
Thank you.
That much in one summer. I also love the colloquial Appleseed banana. That was great. Can you talk a little bit about what you had to do to work with MRSA in your laboratory setting?
So we always work with the bacteria in a biohazard hood. So the hood has a clear door shield that we pull down so that we're not breathing in the bacteria. I also chose to wear a mask, and we, of course, wear gloves to make sure that, and lab coats to make sure that we don't get any on us. We're also working with it primarily in the auger, so I didn't have to touch the actual bacteria.
One more question.
You mentioned that the two compounds you extracted had already been identified. Was that just in a, you knew they existed, or somebody had already looked at them for biomedical context?
Both. So we ran them through databases and found that they've already been identified before, and some of them have been tested in prior studies.
As a follow-on, were they also identified from sponges previously?
Yes. Thank you.
Our next speaker is Ms. Kendall Schlitt from the University of Florida. She is going to present on the health assessment of free-ranging juvenile green sea turtles captured in the Lower Florida Keys, and her mentor was Dr. Annie Page.
Hi, I'm Kendall, and I'm here to talk all things green sea turtles. All right, so why do we even care about green sea turtles aside from, they're cutie pies? They are threatened and endangered throughout the world. Specifically in our area, they're threatened. Something else that's interesting about sea turtles in general is fibropapillomatosis, or FP. FP is a viral infectious disease that causes neoplastic growths. While these growths are benign, they can just be in areas of the turtle that can cause detriments to their survival, especially if the tumors are around the eyes, mouth, or flippers. And this picture of the turtle, I don't know if you can see it, but there's eye, mouth, and flipper, so triple whammy, poor guy. FP has a multifactorial etiology. Studying the etiology of the disease is really important. We do know that there is a strong correlation between FP and clonid herpes virus number 5. Like I said, determining the etiology of FP is really important because that is part of the recovery plans for green sea turtles, as well as documenting turtle health information. Documenting turtle health information is really important in terms of establishing reference intervals. If you're going to go get your blood taken, you'd want something that tells you if it's normal or not. And so that is really important to determine for green sea turtles and wildlife in general. It's also important to document regional differences. Diet, habitat, and stressors all cause differences in health information. So for this project, our objectives included summarizing all health information, as well as documenting the prevalence of diseases. We tested for FP and 2 alpha herpes viruses, clonid herpes virus number 5, and lung-eye trachea virus. We also wanted to establish reference intervals and identify any correlations between health data and disease. One hypothesis that we had was that there would be a difference in blood values between turtles with FP and turtles without FP. These are all the many analytes that we looked at. Morphometrics, so we have size and weight, we have vitamins, hematocrit, biochemistry, which is giving you a little information about the internal organs and overall health of the organism, complete blood cell count, which gives you information about infections, and toxicology, which includes heavy metals. So for the methods, the turtle samples were collected before my time. They were collected during 2019, 21, and 22. That is a picture of my lovely mentor gathering some of the sample. They were hand captured in the lower Florida Keys in a very humane way and returned within, I believe it was 3 minutes. The whole blood, when I got here, we sent off some whole blood samples to Michigan State for heavy metal detection and to the University of Georgia for a quantitative PCR for LETV. In addition, I extracted some blood DNA from the samples and then ran a qPCR for clonid herpes virus number 5 just to round out the data that we already had. We just used Excel to summarize the data. That includes mean, median, range, standard deviation, just your typical statistical summary. Our software was used for just statistical testing and correlations, and then MedCalc was used for interval determination. To determine a reference interval, I only used the healthy turtles. So these are the turtles that tested negative for all 3 diseases. And then this is just a screenshot of what MedCalc looks like, so you can just see what I plopped into the software. To determine a reference interval, like I said, I only used healthy samples. So if there were 20 or more, then hooray. If there weren't, then we could not determine a reference interval. From there, we determined if the distribution was normal. We just used the Anderson-Darling test for that. If it was, you can use a parametric method, which is a percentile method. It's just determining the middle 95% of the samples using the empirical method. And then, if not, you would use a robust method with bootstrapping, and that is done solely in MedCalc. It's an iterative process. In a similar manner, to identify any correlations, we once again had to determine if the distribution was normal. If it was, we used the t-test, and if it wasn't, we used a Mann-Whitney U-test. So these were the reference intervals we were able to establish specifically for green turtles in the lower Florida Keys. As you can see, there are some without a lower limit, and that was just because the data that we had was skewed right, so the lower limit just didn't quite make sense in the context of, health. Oh. So then also these are the significant correlations that we identified, that there was a difference between the distribution of turtles with FP and a difference without FP. So you can see at the top there, packed cell volume was lower in sea turtles with FP than without. The morphometric values were larger, so that means that the sea turtles were bigger. And then the 3 heavy metals were lower, although they are all technically within the trace value limit. And then these are just, again, all the analytes that we tested. These did not have a significant correlation between FP and not FP. So the reference intervals established will contribute to an ongoing health database for green sea turtles. Again, it only applies to the lower Florida Keys aggregation, but it is important to compare it to other green turtle aggregations. So my mentor, again, has done this in Puerto Rico. For the turtles that I studied, we had lower alpha-1 globulins than the turtles in Puerto Rico. Our turtles had higher alpha globulins than the ones in St. Lucie Indian River Lagoon. There were no differences between our turtles and turtles from the Bahamas. But there were quite a few differences between our turtles and Australia. Something to note, though, we didn't test all the same things, so it's only the things that both studies tested the same. And then I just made this little map to show Australia is far. And that's important because, as aggregations are farther apart, they're going to have different values. They have different diet, different lifestyle, different environmental stressors and everything. So that's just why it's so important to have these regional studies. All correlations that we identified did support previous research. We found, larger turtles had more prevalence of FP, and this can be kind of explained with just the life cycle of a turtle. They do tend to return near shore in large groups, and that's when they kind of just start spreading this virus around. The tumors take 15 to, I think, 43 weeks to grow. So that's when we'll start to notice them, is once they've been exposed and had time to develop these tumors. Lower PCV or PEC cell volume was identified in FP positive turtles. Low PCV can indicate anemia. This can be due to infection, blood loss, parasites like leeches, and not eating. I mentioned leeches specifically because not only are they going to be sucking blood, they are a mechanical vector for FP. And then one thing to note, though, in this study, the mean PCV for FP positive sea turtles was not necessarily below the reference interval that we just established. So it's not necessarily anemia that is the problem, but it does indicate a level of immunodepletion. And then we found lower heavy metal concentrations in FP positive turtles. And this just contributes to the fact that we're not quite sure how it goes together. One study found that there were no differences between FP and non-FP turtles and heavy metals. And then another one found that FP positive turtles had higher levels of heavy metals. So just another unknown. As far as future directions go, completing the data set using the stored samples would be good. We had all those analytes, but we weren't able to analyze all of them because we only had 1 year that tested for vitamins, but we do still have all the stored blood samples. So as time and money allows and grants and everything, completing that data set would be great. As permits allow, continue gathering samples from the same aggregation to increase our sample size for all values that we tested for. It would also be cool to see more than just juveniles, so maybe adults. And then obtaining environmental parameters would be great, especially over an extended period of time. It is hypothesized that FP can be caused by certain environmental parameters, just not exactly sure what and how. Replicating the study again for other aggregations is really important to keep getting this regional information, and then focusing our research on blood cell diseases, infection indicators, size, and diet. A big thank you to everyone. We have my mentor, Dr. Page, Wendy, Matt, Adam. Again, I'm sponsored by the Link Foundation. All these other people are lovely partners. And then I wanted to point out this manatee. I got to witness the Marine Mammal Rescue Team rescue the manatee, and that's my dog. Thank you.
So we have time for a few questions. So I have a question for you, and which show my ignorance, but so do turtles kind of form groups and that's where they always live and stay, or do
They
come and
go through phases. When they're younger, they skedaddle into the ocean, and then they stay near shore as they're younger, and then as they get older, they do go out to sea. They do their things.
Nice presentation. I think you mentioned you'd only sampled juvenile turtles. You find this disease is more common in juveniles versus adults, or is there a difference in life stage?
Honestly, I forget. I'm sure I read it somewhere. Once I honed in on the juveniles, I stuck there. But that was just what happened to be sampled, and it's just based off size.
Our next speaker is Ms. Ella Caracazzi. She comes from Florida Atlantic University, and she's going to talk to us on the impact of thermal stress and macroalgal blooms on marine sponges. Her mentor is Dr. Andía Chavez Fonegra.
Thank you so much, and good afternoon, everybody. I'm really excited to talk about sponges today. So, really quickly, coral reefs are really important ecosystems that can be found all across the world, and one really important component of these is sponges. However, in the past couple of decades, we've been noticing an actual increase in events like global warming, which has been causing issues such as elevated water temperatures. Now, this is really critical for coral reefs because this causes mass bleaching and mass mortality events. Now, this causes corals to die off, and because of this, scientists have been actually noting a recent shift from coral-dominated reef ecosystems to more sponge and macroalgae-dominated ecosystems. Now, another phenomenon that has been occurring a lot with elevated water temperatures is the appearance of macroalgal blooms. Now, we've kind of talked about this a little bit today, but when macroalgal blooms occur with the sense of thermal stress, this can have a direct impact on sponges. As you can kind of see here in this photo, when they occur in such abundant mass amounts, they can actually tend to smother organisms that live underneath it. Now, this is a problem in the fact that it doesn't allow sponges to perform their proper ecosystem functions in important roles such as nutrient and water filtration that benefits everything else. Now, another phenomenon that has been occurring as well is actually disease outbreaks, specifically white veil disease. Now, this disease is placed in quotes because it is not a disease in the classical sense, nor is it actually a properly named disease. As you can see in this photo, it's actually only been recorded a few times across the globe, specifically in the Mediterranean and off the coast of France. But what's really unique about it is it seems to be a white microbial mat that covers sponges, but the actual origin of what this actually is unknown, but is actually currently being worked on in the lab to figure that out. Now, specifically, people have kind of assumed that it could be a stress indicator or some sort of what we call sponge dysbiosis, which is when the internal of the sponge actually sort of destabilizes from possibly thermal stress and actually turns in on itself and produces what we can actually see here. So this two main types of sponges that we're going to be looking at that can actually show some sort of sponge dysbiosis is what we call HMA and LMA sponges. Now, this stands for high and low microbial abundance. And as you can see in this graph here, a sponge that is considered an HMA actually has a lot of what we call microbes, or basically little bacteria that is living inside its tissue. Now, this is actually really beneficial because it poses as a great defense system and makes them less vulnerable and susceptible to diseases. But in the case of thermal stress, because there are so many different things living within the sponge, this can actually cause them to destabilize and actually produce sometimes some sort of disease or actual sponge dysbiosis. Now, in LMA sponges, on the other hand, as you can see, there's not a lot of microbes living in their tissue, which can actually make them more vulnerable to things like white veil disease. Now, to kind of put this all together, since there's a lot of moving parts, we end up with what we call the vicious cycle. Now, this has three main components and three main parts. We start with seaweed or macroalgae that exudes actually what we call dissolved organic carbon, that is nutrients quickly reabsorbed by sponges in their tissues, filtered, released back into the water as particulate organic carbon, reabsorbed by corals, filtered, put back into the water, and then reabsorbed by seaweed. That then starts the whole cycle again. Now, with a lot of coral mortality that we're seeing corals coming out of this equation, which is leaving an extra abundance of nutrients that is really creating this positive feedback loop that is giving sponges a lot of nutrients, but also increasing the amount of macroalgal blooms that we tend to see. So what we kind of wanted to look at, that was our main objective, is to evaluate how temperature and macroalgae impact overall sponge health and survival. So this experiment actually took place in Bocas de Toro, Panama. As you can see here, Bocas de Toro is a really small island in the Caribbean, and over here is a close-up of what the island actually looks like. So over here in this yellow square is the Smithsonian Marine Station, where we actually ended up doing this experiment in one of their outwater raceways. And over here in this green square is where the actual collection site took place at a site named Casablanca. So the three sponges that we ended up using were picked for their morphology as well as their local abundance in the area. So our first species is Nephates erecta, which is this really lavender rope sponge right here, which is classified as an LMA that we just went over. Next, we have Cleona deletrix, which is our red boring sponge, also known as an excavating sponge with a unique morphology in which they will actually bore and dig into dead coral and living in the top as well as inside versus a regular Nephades erecta or any other sort of sponge. Lastly, we have our Aplycina califormis, which is this really long rope sponge here, which is actually classified as an LMA. So with these three species in mind, we hypothesize that if they were exposed to thermal stress in macroalgal blooms, they'll actually exhibit poor health and survival overall. So to actually complete this experiment, we had our 3 different species and we collected 5 individuals per each species. Now out of those 5 individuals, we cut them up into 4 pair, a total of 8 fragments per individual. From there, out of 1 pair, we took both of them and stuffed them into mesh bags. But to mimic a macroalgal bloom, one of those pairs was stuffed with a ceramium species of macroalgae, just enough to sort of smother the sponge to kind of mimic what a macroalgal bloom would do. And you can see this indicated by the green dot right here. Now, to sort of mimic thermal stress, we had 4 tanks, 2 with the thermal stress that literature indicates at 32 degrees Celsius and ambient water in the area at 28 degrees Celsius. Now, we had 4 tanks to kind of monitor for any sort of tank effect that might occur in all of the fragments. But from there, each fragment was placed randomly into one of these 4 tanks. And as you can see here, we have 3 heaters in sort of a U shape to keep in mind to make sure that the water was heated consistently and to monitor for any sort of heater bias with the macroalgae. All of them were placed in zigzag forms in the tank. That way, not all of the macroalgae deteriorated right up against the heater. So this experiment was relatively short, only lasting 10 days, with photographs taken each day to monitor for 7 different health metrics, including discoloration, white veil disease, microbial film paleness, and survival. But the 2 that we'll be looking at is survival and white veil disease. So heading into our results, we wanted to look for was the actual health symptoms that each of these fragments portrayed. So as you can see here, we have 2 main plots, our 28 and our 32 degrees Celsius. Now, they're color-coded based on their species with photos here for reference, but as you can see, our blue nary and our purple acal fragments seem to tend to overlap a lot more. But when you look at our Cdel fragments, you can see there's a cluster here, but it tends to be a little bit less on the outside of it. So this kind of indicates that our Cleona fragments seem to show some sort of health symptom that is not overlapping with the rest of them. But it being slightly closer indicates that it still showed some health metric symptoms that the other 2 displayed. However, when you look at our 32 degrees, you can see that they tend to overlap a lot more, suggesting that they actually showed very similar patterns of health symptoms across the 10 days. Now, we actually did another test to discover what specifically was a different symptom in Cleonas, and it actually turned out to be necrosis, as you can see here. So a lot of them tended to show different health symptoms, as you can see with the overlap, but necrosis tended to seem to be the only symptom that showed up in Cleonas, which is actually just the deteriorating of the surface sponge and causing it to turn almost black. So in terms of their overall survival with the multitude of different factors and variables we have 4 plots with the x-axis showing the experiment as we go from day 0 to day 10, and on the y-axis our mean survival proportion in percentages. So as you can see in our 28-degree tanks, Cleonas actually showed a 100% survival rate, indicating that macroalgae had no effect on the mortality at all with this temperature. However, when you tend to look at acal and nary, when paired with macroalgae, they showed around an 80% survival rate. However, fragments paired with that macroalgae showed 100% survival. Now, this pattern completely changes when you throw these into our thermal stress tanks at 32 degrees Celsius. As you can see, Cleona still once again has 100% survival rate, which is really interesting and likely due to their excavating lifestyle. However, when you tend to look at our nary fragments, you can see it drop off pretty early. When paired with macroalgae, it had over a 70% chance of survival. However, when you get to them being paired with no macroalgae, you only have a 60 to 65% chance of survival, suggesting that macroalgae might be some sort of buffer. Acals had the most mortality out of all 3 species, reaching from about 20 to 25% when paired with or without macroalgae. Now, in terms of the frequency of white veil disease, you can actually see, once again, a big jump in our thermal stress. So it's very similar with our y-axis being a proportion of white veil occurrence and our days 0 through 10 on the x-axis. So as you can see with our macroalgae, no macroalgae, and 28 degrees, Cleonas actually showed more of a white veil occurrence, over 40% of fragments when paired with macroalgae, and around only 40% without macroalgae. However, when you look at our acal, you can see that there's a lot less which in terms of their survival was a lot worse, but they seemed to do better without showing white veil, showing only just under 20 and then jumping down to 10% for with and without macroalgae. Now, NARI tended to show actually no sense of white veil occurrence in these tanks, whether they were paired with or without macroalgae, which also suggests that there might be something, but there might also be not. Now, when you tend to look at our thermal stress tanks, you can see there is a big jump in the white veil occurrence of over 80% of sea delta fragments showing any sort of symptom of white veil. Now, when you tend to look at it without macroalgae, only about half or 40% of those fragments showed symptoms of white veil. Now, this completely shifts from 28 to 32 with ACAL showing over 70% and 80% of fragments showing an occurrence of white veil disease. And when you look at NARI, only 40% with macroalgae and around 45% with macroalgae showing some sort of white veil disease. Now, what does this kind of mean? Because there's a lot of things going on. So basically, this all shows that there's not one general pattern that can be observed for all three species, but rather these are more species-specific responses. Now, ACAL seemed to generally do the worst and seemed that they were the most sensitive to thermal stress and macroalgal blooms in general, which makes sense with the fact that they are an HMA sponge and tend to have a lot more microbes in their system and show a lot more dysbiosis. Now, in terms of our CDEL, they showed a lot of necrosis that you can see here, but they had no mortality, likely to the fact that they can bore into dead coral, but they seem to be really sensitive to white veil disease, especially for the fact that they're an LMA. Now, our other LMA showed a completely different pattern out of this, where NARI had overall the lowest mortality and disease rates, with macroalgae being some sort of buffer to thermal stress. Now, in conclusion, thermal stress and macroalgal blooms seem to impact the survival of our HMA sponge more than our LMA sponge and increase the disease frequency in our Cleona deletrix. However, this pattern completely shifts when you see that it actually increased the survival and decreased the disease frequency in Nephates erecta, with macroalgae indicating some sort of a buffer against thermal stress. Now, before quickly finishing, I want to give an acknowledgement to everybody on this list and everyone who showed up today to listen to this presentation. I really appreciate it. And a big thank you to the Gale and the Link Foundation for providing the funding for this research opportunity to be possible. Are there any questions?
Do you think that the prevalence of white veil on the flooring sponge could be because of the structure of it itself, since it has a larger surface area?
That's a great question. I would definitely say that it's possible, but we still saw a wide occurrence of white veil disease in our other two species. So the difference with the Cleona versus the other two is because it's actually on the dead coral. It can grow on the top, but since it also bores into it, white veil actually doesn't appear on the inside, which is why when we cut into them at the end of the experiment, we were actually able to determine that there was no mortality rate. So even though you physically look at them, there's still no mortality, but when you look at sort of a regular rope sponge, for instance, the appearance of white veil is able to go basically entirely through the sponge. And then at the end, when you can see it, I think it might be able to see here. Yeah, you can see that it basically just, you can see the entire skeleton of the sponge, if that answers your question at all. Yeah?
So white veil just been located in the Mediterranean?
So the first occurrence of white veil disease, there was two papers originally. There was one in 2011, which was the one in the Mediterranean, and then there was one a year later in 2012 off the coast of France. There's been a few other occurrences, like the one that NOAA has discovered. That one was taken in 2016. And then there is a lab student currently who is also working on this white veil disease and actually did her thesis on this the year before, so our patterns ended up being very similar. But there's not a lot of like recorded observations, and because there's no label for it, people when they go out don't really understand like what to look for and how to look for it. So there's not a lot of like records of like what it actually is or how common it is.
On your presentation for the white veil disease, it looks like it increased to its highest around, it was probably 6 or 8 days, and then decreased again at the end of your experiment.
Is that due to mortality or?
Yeah, so a lot of the time what happened was there were 3 kind of different severity levels when it came to white veil disease. So when white veil disease would occur, it would, for the most part, once you recorded the first symptom of it, by the time that you reached a day or 2 later, it was incredibly severe to the point where it was basically just like a white sheath over the entire bag, and you don't really want to touch it because then that could probably spread it more. But then sometimes with the mortality, because as you could see like in the Navati skeleton, it's like there's nothing for that white veil to hold on to, but there was also the rare occasion, looking back at the photos, where white veil would become severe, and then before the end of the experiment, it would actually decrease in severity and the sponge would still survive. So there were both patterns that seemed to show. Thank you.
Our next speaker is Miss Isabella Holden. She comes to us from Florida Atlantic University, and she will speak on ocean acoustics, educating our community about underwater sound. And her mentor is Dr. Gabrielle McHenry.
Good afternoon. My name is Bella Holden, and this summer I had the pleasure of working with the Education and Outreach team, Dr. Gabby McHenry, and the Fisheries Ecology and Conservation Lab to educate the community on underwater sound. The underwater world is alive with sound generated by marine animals, environmental events, human activity, and even predator-prey interactions. These sounds play a crucial role in how marine species navigate, communicate, and survive. Here at FAU Harbor Branch, the Fisheries Ecology Lab is dedicated to exploring and understanding the complex dynamics between finfish, elasmobranchs, shellfish or mollusks and crustaceans, and their ecosystems. The lab is currently using hydrophones or underwater microphones to study durophagus or shell-crushing predators and their interactions with shellfish, which are vital for maintaining water quality, supporting food webs, and stabilizing shorelines. By identifying the unique sounds of shell fracture, researchers can estimate predation rates and assess ecosystem health in real time. In addition to this, the lab uses tagging and stationary monitoring to investigate how predators move through different habitats, how environmental conditions influence their foraging behavior, and what this means for the survival of a key prey species. With that being said, the goal of my summer project is to educate a diverse audience on underwater acoustics, the relationship between durophagus predators and shellfish, and corresponding FAU Harbor Branch research. I accomplished this goal via 3 objectives: the integration of a hydrophone into boat programming aboard the Discovery, the development of a new exhibit for the Ocean Discovery Visitor Center, and direct public education by showcasing at community events. My summer project is deeply rooted in research currently being conducted by the Fisheries Ecology Lab, which led to frequent collaboration with their team. During our initial meeting, they generously lent me a hydrophone and handheld recorder, which I used to conduct several excursions around campus and document the common sounds of the channel. I also had the opportunity to join the lab in the field, which gave me a clear understanding for their research activities and allowed me to take photos for my project. So objective number 1, integrating a hydrophone into programming aboard the Discovery. For those of you who aren't familiar, the Discovery is a 36-passenger pontoon boat used in Harbor Branch's public tours and field trips. Each row in the boat is equipped with weatherproof screens, allowing the guides to pull up media that accompanies the dialogue. The major goals of these boat programs is to demonstrate and inform visitors about the tools and technologies used by scientists at Harbor Branch. One major piece of technology used on the boat is the ROV, which lets guests visually explore the underwater world. A hydrophone, on the other hand, would offer a different perspective, allowing them to listen to it, making it a perfect addition to the experience. I joined these programs to better understand the flow of the presentation and identify the best time to incorporate underwater acoustics. I then began adding to the pre-existing scripts for the boat programs, making sure to include the hydrophone demonstrations, channel sounds, and ocean acoustics research. My next step was to connect the recorder to the overhead speakers of the Discovery so that passengers would be able to hear the underwater sounds live, which resulted in various technology purchases. I was then able to add relevant content to the PowerPoint slides, including an image of the recording equipment and an audio library of 4 animal sounds guests may hear after hydrophone deployment. The sound bites include dolphin whistles, manatee squeaks, porkfish grunts, and snapping shrimp. I also developed a document that outlined how to troubleshoot, use, and clean the hydrophone and recorder. My next step was to order a hydrophone and handheld recorder of our own, which resulted in the purchase of a variety of accessory tech supplies, as well as materials for fashioning a case to protect the transponder. I then created a protector for the transponder out of foam inserts and a plastic case made for earbuds. I was now able to test the updated programming on field trip groups, which allowed me to refine my slides, script, and standard operating procedure. I then trained all the staff involved in the boat programs and joined the first boat trip after this to lend my knowledge of the technology and the sounds recorded. To evaluate the impact of this new addition to the experience, we tracked participation in field trips from July 10th to the 23rd and surveyed attendees with 2 questions. During the presentation, students responded by raising their hands. In total, 142 individuals experienced the hydrophone and underwater sound segment, though only 120 were present for the survey portion. The questions asked were, do you know what a hydrophone is, and do you remember what a hydrophone is? The former was asked before beginning the underwater sounds presentation as a benchmark, and the latter was asked after to gauge retention. The number of hands raised for each question was then recorded. Out of 120 participants, approximately 20.8% said they knew what a hydrophone was before the presentation. After the presentation, 81.7% said that they remembered what the definition of a hydrophone was. Therefore, the number of students familiar with the device increased by 60.9%. To evaluate the impact of underwater sound experiences on public boat tours, participants were given the option to fill out a survey once they deboarded. 48 people experienced the hydrophone and underwater sounds presentation on a boat tour, and of these 48, 22 were offered a survey. Of these 22, 7 completed the survey. They were asked the following: did you enjoy the hydrophone demonstration? And do you have any feedback or suggestions as to how to improve the hydrophone demonstration? 100% of guests surveyed said they enjoyed the hydrophone presentation, and most guests said they were pleased with the experience as is. In response to the feedback and suggestions prompt, some participants stated that they felt the hydrophone would complement the ROV experience well. We also received suggestions to add information about the hydrophone's range and how sound moves underwater. We will take that feedback into account as we continue to update the experience. Some notable survey responses include, I was excited to hear what was going on under the water, and learned about porkfish. They snort like pigs. Since this new segment will be a permanent part of the boat program, we can expect that approximately 3,000 people each year will experience the hydrophone demonstration and underwater sounds presentation. Objective number 2, developing a new exhibit for the Ocean Discovery Visitor Center. The Visitor Center is a building on campus that is open to the public and features self-guided exhibits on current research taking place at Harbor Branch. One of these exhibits is titled Marine Ecosystem Health, and I created a display within this space. My first step in accomplishing this objective was to interact with this original exhibit to understand how mine should look and operate. The previous display included a table and a touchscreen computer. I then began developing my interactive module titled Audio Waves. I used PowerPoint to create the module, where I hyperlinked slides together so that, when in kiosk mode, visitors could click through different slides regarding marine soundscape, measuring underwater noise, and pertinent Harbor Branch research. The module also had sound bites of environmental, anthropogenic, and animal sounds, as well as footage acquired from the Fisheries Ecology Lab. I then tested the audio on the exhibit display and purchased 3 headphones and a 2-way splitter to allow visitors to listen to various underwater sounds. I also created a document that outlined the cleaning procedure for the headphones and display, resulting in the purchase of a pack of microfiber towels. My next task was to build out the research section of my presentation, which led to the purchase of an external hard drive that allowed me to download a plethora of media from the Fisheries Ecology Lab. Once the digital component of my exhibit was complete, I addressed the physical component, the props, their descriptions, and the headphone display. I added a stone crab claw to the exhibit and updated the associated placard to reflect this addition. It will now include a description of the cow-nosed ray and stone crab's adaptations for preying on hard-shelled animals. The revamping of the display resulted in the purchase of small plastic stands to improve the presentation of the props, as well as headphone mounts. I then tested the new display on the education and outreach staff. The Audio Waves module and display materials were then passed on to the Education and Outreach Department leadership and the Fisheries Ecology Lab for review and approval. The new exhibit is expected to debut within the coming months. 5 employees of the Education and Outreach team were given a feedback sheet to complete as they interacted with the Audio Waves module. They were asked to provide feedback on the following regarding each of the 3 sections: the media used, the maneuverability of the presentation, and the comprehensibility of the material. They were also asked to estimate guest reception and provide ideas for improvement and an overall rating of the exhibit. The Education and Outreach Department will use the feedback received to finalize the display. The majority of participants responded positively to the media used in each section of the presentation. They also reported that the content was easy to navigate and understand. 100% of staff surveyed believed that guests would enjoy the exhibit, while 60% felt that the presentation was ready to be debuted to the public. Overall, the module received an average rating of 9.2 out of 10. Once this new exhibit is officially introduced to the Visitor Center, we can expect that it will be seen by 4,000 people every year. Objective number 3, direct public education. This summer, I was able to attend 2 outreach events, the Fort Pierce Saturday Farmers Market and World Oceans Day event at the St. Lucie County Aquarium. In addition to the typical festival materials, I brought my laptop, fitted with headphones and complete with a PowerPoint displaying underwater sounds, and a cow-nose raised jaw, a shell, and an acoustic tag. Therefore, attendees at these events had the opportunity to listen to underwater sounds and learn more about the animals that make them. Metrics were taken by recording the number of people who visited our table on a handheld tally counter. A total of 33 people visited the Harbor Branch table at the Farmers Market, and 37 people visited our table at the World Oceans Day event. In total, 100 people were introduced to durophagy and underwater sounds at outreach events this summer. If my educational tools continue to be included at tabling events, we can expect to engage thousands of members of the community with fisheries ecology work and inform them about marine soundscapes. The Fisheries Ecology Lab at Harbor Branch plays a key role in protecting marine ecosystems and supporting sustainable fisheries. By studying fish populations, habitats, and environmental changes, the lab provides vital data for conservation management efforts. This research helps ensure that the health of both marine life and coastal communities. It helps ensure the health of both marine life and coastal communities. My project was created to share this important work with the public, supporting FAU Harbor Branch's mission to promote ocean literacy by connecting people of all ages to cutting-edge marine science. With a new exhibit planned for the Visitor Center, updated boat programming already in place, and fresh educational tools for outreach events, the materials I developed this summer have the potential to reach over 10,000 guests annually for years to come. Thanks to this internship and the experiences I've been lucky to have here at Harbor Branch over the past 6 months, I have been offered a job at SeaWorld Orlando as a zoological specialist in aviculture. After SeaWorld, I hope to attend graduate school in pursuit of my master's in biology. But there are other people I must thank first before wrapping up the final task of my internship. I'd like to thank Dr. Gabby McHenry and Jennifer Cutter for their guidance and feedback throughout this experience. I would also like to thank Courtney Brooks, Tracy Griffin, Sam McGuire, Callie P., Joey Story, Lauren Tracy, and Juliana Valmera Fernandez for their kindness, support, and constant willingness to help. They were always eager to offer feedback, serve as the first set of eyes on my work, and lend a hand whenever I needed it. And a big thank you to Captain David Huberger for always giving me the time and space to explore and complete my work on the boat, and to Jessica Carvalho, Maddie DeBest, and the Fisheries Ecology team for lending me their media and expertise. I'd also like to give a big thank you to the Link Foundation for sponsoring my work this summer, and to Dr. Hannah Sack for pioneering the Semester by the Sea program, without which I would not have been able to accomplish all I have in the past 2 semesters. Thank you to all the other staff who helped make my internship successful, and with that, I will now take questions. The coolest thing was probably dolphin whistles, which I got more often than I thought. Those were so cool. But I also heard Chris Stapleton's Tennessee Whiskey over the hydrophone one time, so that was pretty interesting. And did you have a second question? I'm sorry. Just watching the kids on the field trip, their faces light up, and them get quiet and focus and like tune in to what was playing above the speakers, and then they go, "That's the porkfish," or something like that. I was like, oh my goodness, thank you, you're paying attention. Anybody else? That works for me. Thank you, guys.
So that presentation concluded our afternoon session. So before we do concluding remarks, I wanted to ask you to give a round of applause to all our presenters because they did a fantastic job. I also wanted to ask you to please thank Mr. Dylan Bono, who has been our IT specialist behind the curtains, making sure that everything went smoothly, and he did a fantastic job. So thank you, Dylan. I also want to thank Victoria Kidd, Jessica Pettingill, and Kathy Rosmel, because they helped us a lot to make sure that everything went smooth today. So thank you.
I guess, we can't believe that 10 weeks are over. What do you guys think? 10 weeks? Did it seem like 10 weeks? It did. Anyways, we really thank the interns for coming, and we just once again want to thank the sponsors. Without their support, this program would not happen. So that's the Link Foundation, the Gertrude E. Skelly Charitable Foundation, and the Harbor Branch Oceanographic Institute Foundation that funds the Maryland Link internship. Trying to think what other things. I wanted to call out all the people in our research services who helped with all the paperwork to get you guys here, and hopefully helped, they helped us, and all of the mentors. I want to thank all the mentors who spent their summers, and all of their staff and other students and graduate students who helped hopefully make you guys have a really good summer, or help make that a great summer and a great experience. Are there other people who are we not thanking? There might be other people, but if we don't remember you, we still thank you
for your help.
But anyway, so with that, I guess one more thing for the students: get those reports into us by tomorrow at 5 p.m. Just electronic copy. That's all we need, and of course, give a copy to your mentor. They want to have a copy of your report. So I think that's it. So let's thank everybody for coming, all the wonderful talks. You guys are one of the best groups I have ever seen in terms of your talks, and every year it gets better. And so this was a fabulous day. So thank you.